Hi everyone. Happy morning. Happy morning. Oh. Where are you? Where are you? Hi people, can you see me, hear me? Hello? Hello, hello, hello. Hi everyone, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma and thank you so much for sharing your love. Hello, how are you all doing? Good morning, good morning, good morning. Good morning. Thank you so much for this amazing support. Thank you so much for watching the lecture for 11 hours, near about 40 minutes. I'm really grateful and keep working hard. Trust me, NEET 2024 is yours. Hera? And why were you thinking that I'm not in the chat? Yeah, I was there in the chat. I was there in the chat. Now your eyes are red. I'm tired as well, no? So how are you? How are you? How are you? Quickly share the session link with your friends. Let's start the question practice. So whatever we have covered yesterday, today we are going to practice that. These sessions are amazing, bache, because you are not just getting the theory part, you are also getting the MCQs, and that too are very good, right? That questions will help you to understand the concept in a better way. So tell me, how are you? How are you? And why were you saying that I was not there in the chat? Yeah, I was there in the chat. I was there in the chat. Hello, hello, hello. And see this, see this, see this. Thank you so much for your comments. But I am little upset as well because very few students, right, they type the comments and saucy. Don't worry, we'll talk later. So thank you, thank you, thank you, Varsha, Sethi, Yuva, Anjana, thank you, Kalai, Monisha, Bharani, Art Messi, thank you guys, thank you, thank you, thank you, thank you so much for this, and waiting for more comments here. Okay, so revise this, and whatever you are feeling about the lecture, do let me know in the comment section. Okay, and yes, is it done? Everyone tell me in the chat section, is it done? Is it done? Yes, this enrollment in the mock test in the AIMT, is it done? And you know what? Today, after the lecture, I have planned something very amazing for you. You people are going to get a very good video. That will be an eye-opener for all of you, right? So today, after this class, within 30 minutes or 40 minutes, I'll be posting a video. Okay, I'll be posting a video. And actually, I was having my breakfast. That's why I started, late. Okay, so chalo, let's start the question practice, and I hope you all, right, you all are ready to give your test as well, because here you are going to analyze your marks, you will get the ranks, you will analyze your paper, you will improve your mistakes. And today, right, I want all of you, see, it's biology, right? So there is no need, right, to practice the question for 7 hours, 8 hours, 10 hours. Okay, you know that we have to see the question, we have to answer the question, right? We have to see the question, we have to answer the question, and that is the best time to increase your marks as well. I'll try to keep my phone with me, okay, and I'll ask you the questions as well. So what you have to do, students, what you have to do, you even need to count your score as well, right? So make sure you have your calculator, phone, or on paper also you can type it. So you will tell me about what? About your marks as well. So after every 20 questions, this is what I'm going to ask. And see, holy vibes. My phone cover is totally changed. It's all holy color. See? Okay, chalo, let's get started. So guys, quickly hit the like button as well, and here we go. And here we have our first question. Okay, so we have to see it, we have to answer it. See it, answer it. So be ready, be ready, be ready. The first question is very simple. Who proposed the central dogma in molecular biology, which states that genetic information flows from DNA to RNA to protein? So in this one question, we have two questions, isn't it? We have two questions in this one question. It is also telling us about the central central dogma of information, isn't it, people? It is also telling us about the central dogma of information. And yes, you are right, the solution is 3. The solution is 3. The speed is fine, guys. The speed is fine. The speed is fine. Yes? Or you want me to be very quick? So central dogma of information is saying DNA will form RNA, RNA will form protein, that is transcription, that is translation, and yeah, it is given by Francis Crick. So without making any mistake, answer the questions and trust me, right, and mark my words, right after NEET 2024, definitely open this lecture. Open this lecture, you will say, yeah, seriously questions are from this lecture only when it comes to the zoology class 12. Next question, if in DNA double strand the cytosine is 40%, what will be the percentage of adenine? I know you can answer it right now. I know you can answer it right now. So I'm sitting here. You please answer the question. Guys, be quick. Be quick. So I'm not going to disturb you much today, right? So we will finish these questions in till 4 or 5, okay? So that you will get time for practice. So cytosine is 40%. Cytosine is 40% means guanine will also be 40%, right? What is it? What is it? Chaga fruit. Okay, so 40, 40, 80, we are left with 20% of the bases. So divide it equally in between the adenine and the thymine. So you got your answer. The answer will be 4. The answer will be 4. Now let's move to the next question. What is the haploid content of DNA in a typical human cell? What is the haploid content of DNA in a typical human cell? The haploid content is asked, not the diploid one, not the diploid one. Again, it's very easy. 3.3 into 10 raised to power 9 base pair. Okay, so let's move to the next question. Who first observed that for a double-stranded DNA, the ratios between adenine, thymine, guanine, cytosine are constant and equals 1? Again, a very simple question. Again, a very simple question. So see how fast we are covering everything. So our Arvind Chargaff, right? Right, our Mr. Uncle Arvind Chargaff. Okay? Our Uncle Arvind Chargaff. He was the one who said it. Okay. Next. In a typical nucleus, the euchromatin is said to be. Guys, are you feeling confident? Are you feeling confident? The euchromatin is said to be. It is said to be exactly transcriptionally active. Transcriptionally active. Next question. The unequivocal proof that DNA is the genetic material came from the experiment of. Just write down the name of the scientist in the chat section. Quick. Just write down the name of the scientist in the chat section. Quick. I'll have my breakfast also. Write down the name of the scientist in the chat section. See, I'll give you one trick. We remember it as Hershey and Chase. Okay. And they are going to ask it like this also. But if by chance, if they mention Alfred and Martha, please be alert. Maybe they will mention the first name. Okay, NTA can do anything, isn't it? NTA can do anything. So please make sure you don't miss it. It's first, that is your answer. Now, Tyler and the colleagues proved that the DNA in the chromosomes of Visia fiber also replicates semi-conservatively using, yes Vishnu, using, yes. So guys, if you are going to give the correct answer, you can have one chocolate, one burger, one pizza from my side. Okay, so go to the local shop, tell them that I told you to buy it, right? And they will mention it in my account. Okay? Go have your burger, have your pizza, whatever you want to have, the chocolate, whatever you want, have it there. Okay? Just take my name there, okay? And see, if they don't know me, then just show them the video and tell them, watch it till the end. Okay, ask them to watch the video till the end. Okay, and tell them to mention the comment as well. And yes, how can I forget? Ask them to join AIMT also. Okay? Ask them to join AIMT also. So basically you will promote me. You will promote me. Chalo. So here we go. So what should be the answer? It is radioactive thymidine. Radioactive thymidine. Now I want the answer of one question. What is the difference in thymine, thymidine? What is the difference in these two words? Thymine and thymidine. Answer my question, answer my question, answer my question. What is that? Saucy, very good. This is what you have to do. Okay? Go to them and tell them, yeah, you don't know her. She's the one. So in her account, mention everything. So whenever I'll visit that place, I'll pay them. Okay? I'll pay them. Exactly. Thymine is the nitrogenous base. Thymidine is the nucleoside. Excellent. Next question. The precursor of mRNA in the eukaryotes is transcribed by. The precursor of mRNA in the eukaryotes is transcribed by. It is transcribed by. It is transcribed by mRNA. Precursor of mRNA means we are talking about hnRNA, heterogeneous nuclear RNA, and definitely it will be RNA polymerase 2. Okay, next, a change of single base pair in the gene for beta globin. That question is from Principles of Inheritance and Variation, but I believe you can answer it because you people are not smart. You are very smart. है ना? You people are not smart. You are very smart. Quick. Quick. Exactly. They are saying the change of single base pair in the gene for beta globin that result in the change of amino acid from glutamic acid to valine. It results into a disease called sickle cell anemia. So it is autosomal, na? Autosomal recessive, right? Autosomal recessive, isn't it? Okay, next. Which of the following is a ribozyme in bacteria? Which of the following is a ribozyme in bacteria? Yes, which of the following is a ribozyme in bacteria? Yes, ribozyme when RNA is acting as an enzyme. So it is 23S ribosomal RNA, and you know that it will catalyze the formation of peptide bond, right? It will catalyze the formation of peptide bond. Next, the blind approach of simply sequencing the whole set of DNA, that whole set of genome that contain all the coding and non-coding one. It will be assigned as, be quick. We are not going to take much time to solve all the 500 questions, bache, right? To solve all the 500 questions, be quick. What is it? It is the sequence annotation. It is the sequence annotation. And EST means when only mRNAs are getting sequenced. Okay, only the expressed genes are getting sequenced because gene has expressed itself in the form of mRNA. Now, which is wrongly matched with its specificity? Which one is wrongly matched with, incorrectly matched with its specificity? Quick. You know the answer, guys. Quick. Quick. Wrongly matched. YAC. Is it a type of vector? Yes. SNP, single nucleotide polymorphisms? Yes. How do we pronounce it? SNPs they are, right? Human largest gene, the gene for dystrophin. Chromosome 1, 231 gene? No. It is having the maximum genes. So solution is 4th. Next, find out the incorrect match. Incorrect match again. So make sure you read these words carefully. You have to figure out the incorrect match. Incorrect match. Right? Yes. What should be the answer, guys? Speed. Your speed is very less, yeah? Quick. You know the answer. Yes, it is euchromatin. Inactive? No, it is active. It is transcriptionally active. Next question. What is the basic concept of transformation experiment of Griffith? Yes, bache. Transformation experiment of Griffith. Basic concept. Read all the options carefully. R2 strain, it will get transformed into S3 strain. Now, DNA component is. Yes, DNA component is. Guys, make sure you hit the like button. Make sure you join AIMT as well. So, DNA component is. DNA component is, it is nitrogenous base. Is it the ribose sugar? No, deoxyribose sugar. Okay, and the phosphate. So which type of bond is not present in DNA? The word is not present. The word is not present. Yes, which type of bond is not present in DNA? I know you know the answer. I know you know the answer. Yes, exactly, the disulfide bond will not be there. Now, nature of histone protein is, 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 exactly, it is basic and positively charged. So which amino acids are present there? The arginine and lysine. Okay, which amino acids are present there? The arginine and lysine. Arginine and lysine. Is that clear? Is that clear? So K and R. Next question. Match column 1 with column 2. My favorite kind of questions. Match column 1 with column 2. My favorite kind of questions. Quick. It's my favorite. Column 1 with column 2. Helicase. What that helicase will do? The opening of DNA? Yes. Okay. The topoisomerase. Remove stress at the time of unbinding. Ligase. The joining of. Okay. Done. So ligase. Don't you think that ligase will be the one that will join the Okazaki fragments? Yes. Don't you think so? Don't you think so? It should be the one that will join the Okazaki fragment. Of course. Oh, not of course. Ruko. Ligase is the one that will ligase the Okazaki fragment. And then your DNA polymerase 3, what that DNA polymerase 3 will do? It will join the nucleotide. So helicase will go with, one will go with D. Okay. So cancel this option, cancel this option. 2 will go with C. Okay. 3 will go with B. I told you already. Okay? Right? So don't get confused here. Dekho, Okazaki fragments, they are like this. Okay. So ligase will come to join sugar phosphate backbone. Ligase will come to join sugar phosphate backbone. That is what you need to remember. Is that clear? Next question. The base sequence in, yes. Yes. This is what I explained in the last class. This is what I explained in the last class. Right? Right. This is what, this is not incorrect. Opening of DNA is done by helicase. No, so one will go with the, what's the confusion? Okay, the base sequence in one strand of DNA is this. Then what will be the sequence of bases? No, this is not the one. I was talking about the transcription. So you tell me about this. Ki one strand is having this sequence. What will be the sequence of base in another strand? I think you know that. I think you know that. TT, CG, AT, G, G, it's C guys, it's CC. They go T, T, C, G, A, T, G, G, C and A. It should be 2. Second. Next question. RNA is more reactive or unstable to DNA due to the presence of? RNA is more reactive or unstable to DNA due to the presence of? Due to the presence of? Exactly, very good. The first is the answer. OH group on second position of ribose sugar. Next, which was the first genetic material on earth? On earth. First genetic material. Don't just read the genetic material. First genetic material we are talking. Of course, Mr. RNA. Next, which of the following synthesize a repressor protein? Which of the following synthesize a repressor protein? The word is repressor protein. The word is repressor protein. Very good. Regulatory gene is the one. And let me tell you, it's not mandatory that always it will synthesize the repressor protein. Your regulatory gene, your regulator gene, it can also synthesize activator. But if it is synthesizing the repressor, it is the negative regulation. What's that? It is the negative regulation. What's that? It is the negative regulation. Now, right, next question. DNA fingerprinting involve identifying differences. Right? Chalo, I'll post a video to explain the DNA fingerprinting. Okay? Yesterday we missed it. I thought in Biotech I will cover. Then I just forgot. So don't worry, I'll post a separate video to explain it. Okay? Okay. So DNA fingerprinting involve identifying differences in some specific region. That regions are, it's a neat question, exactly repetitive DNA. Next question, how human genome consists how many number of genes? What was estimated and what we got? What was estimated, what we got? It is 3000. So do not get confused. Oh, it is 3000 or 30,000 on an average? You tell me, is it 3000 or 30,000? We are talking about the genes in the humans. Is it 3000 or the 30,000? Quick. Human genome contains genes somewhere in between. Somewhere in between, what should be the answer? 3000 or 30,000? It is not exactly 30,000, you know that? It is not exactly 30,000, do you know that? Right? But it is a PYQ. Students, it is a PYQ. It is not exactly 30,000. It is somewhere in between 20 to 25,000. Right? But as per all the options that are given here, we need to mark the answer. Okay? As per that, we need to mark the answer. They are asking number of genes, not the basis. Right? Not the basis. Next, which statement is not correct? Right? Incorrect statement. Incorrect statement. Exactly. Incorrect statement. Which one is the incorrect statement? Quick. Which statement is the incorrect one? Which statement is the incorrect one? Quick. I think you all can answer the question. Okay. Incorrect one. So amino acid attached with the acceptor end of tRNA. Okay, acha. ठीक है. Release factor binds to the stop codon for terminating it. All 64 codons code for amino acid in splicing introns. Obviously this is not right. So 64 minus 3, 61 codons are going to code for the amino acid. That's the point. Okay, and here you know what we have to do. See, they go genes, number of genes will be 30,000, but on an average 3,000 bases are present in a gene. Right? So don't get confused here. On an average 3,000 bases are present in a gene. Right? But yeah, number of gene is 30,000. Now next, let's move to the next question. Which statement is not correct? Again, the incorrect kind of statement. Which statement is not correct? Which statement is not correct? Which statement is not correct? You know the answer? You know the answer? DNA replication takes place in 3' to 5' direction. No, I told you always it will be in 5' to 3' direction. Next, column 1 with column 2. Column 1 with column 2. Yes, go for it. Go for it. By just looking at one option, you can answer it. Yes, that's the beauty of, right? That's the beauty of match the column kind of questions. Exactly. ये देखो ना, Hershey and Chase. Use of this 3 with D. You got your answer. है ना? Simple. You got your answer. है ना? Okay. The next question. Next question. Next question. DNA as an acidic substance was first identified as name as substance present in nucleon by. Yes. DNA as an acidic nucleus was first identified and named as substance present in nucleon by Frederick Mesher. Next question. The distance between a base pair in the helix of DNA is approximately. What will be the distance? The distance between a base pair in a helix of DNA is approximately. They are saying the distance between a base pair in the helix of DNA, but between a base pair. Yes, so it will be 0.34 nanometer. Okay, next question. The histones are rich in basic amino acid residues like, so this question was there in NEET 2020, this same question was there in NEET 2021 or 2022 as well. Okay. Okay, so it's a very important question. So obviously it is lysine and arginine. It is lysine and arginine. Next question. Which of the following enzymes inhibit transformation during the work of Avery, MacLeod, McCarthy on the biochemical characterization of transforming principle? The question is, which one is going to inhibit the transformation? And you know the reason as well, right? So it is DNase. Why? Because it will digest DNA. So if there is no DNA, there is no transformation. Next question. Presence of which of the following nitrogenous base at the place of uracil of RNA confers additional stability to DNA? Confers additional stability to DNA. Very easy. So it is thymine, which is also known as 5-methyl-uracil. 5-methyl-uracil. Next, the structural gene in a transcription unit in bacteria or prokaryote is mostly, right? The structural gene in a transcription unit in bacteria or prokaryote is mostly, exactly, polycistronic, right? That mRNA can give many proteins. It will be polycistronic. Next, some amino acids are coded by more than one codon. Hence, the codon is said to be, amino acids are coded by more than one codon. Exactly, it is the degenerate. This property is degeneracy and it is due to the wobbly base, right? So first two bases, they will follow the Watson and Crick pairing, and the last base, it can pair with any other base, okay? Any of that four bases. Next question. The secondary structure of tRNA looks like, the secondary structure, right? The secondary structure of tRNA looks like, it looks like, yes, it looks like, exactly, clover leaf. Next, no, the tertiary one is inverted L. Okay, when it is the tertiary structure, 3D structure, then it is the inverted L. No, right? So secondary one, it will be clover leaf like only. So that is the correct answer. Next, which of the following enzyme is primarily responsible for hydrolysis of disaccharide lactose into its monomeric units, right, in the lac operon? Very simple, very simple. It's very simple. Exactly. So it will be beta galactosidase. Okay, it will be beta galactosidase. It will break lactose into glucose and galactose. Okay, so tell me, lactose is a reducing sugar or is it a non-reducing sugar? What kind of sugar it is? Is it a reducing sugar or non-reducing sugar? Reducing sugar or non-reducing sugar? Lactose. The reducing one. Okay, the reducing one. Next question. Polymorphism, that is variation at the genetic level. It arises due to variation at the genetic level. It arises due to, yes, due to, it's a PYQ. So obviously, variation at the genetic level. See, I know when we see this repetitive DNA and satellite DNA, we think that they are the one, right, which are responsible for the variation. But ultimately, when it comes to the variation, are mutations, right? They are responsible for that variation. They will result in the in having that single nucleotide polymorphism and all that things. That's why the answer is 3. So do not get confused. It's a tricky one. Okay, it's a tricky one. Next question. Next is sequence of 3 nitrogenous base of RNA can function as, right? The sequence of 3 nitrogenous bases of RNA can function as, quick. It can function as, quick. Exactly, codon, right? Just say AUG will code for, and I hope you all have checked the video in which, you know, I have explained the genetic code with the tricks. Make sure you check it, okay? Make sure you check it. So it is AUG. It is AUG. Clear? Okay, the next one. Which statement is not correct? Yes, which statement is not correct? Quick. Not correct. Of course, it's very simple. AUG codes for methionine amino acid. It is the correct one. In humans, less than 2% of genome codes for protein. Correct one. Primer is formed by primase. Yes. DNA polymerase is the main enzyme for RNA synthesis. No, for the DNA synthesis. Okay, for the DNA synthesis. So this one is our answer. Next question. In RNA, which type of nitrogenous base is not present as a structural component? Right? Which one is not present as a structural component in the RNA? Quick. Which one is not present as a structural component in the RNA? Of course, 5-methyl-uracil. Why? Because 5-methyl-uracil means we are talking about the thymine here. Okay. So second is the correct answer. Now next one, again match the column kind of question. Very simple it is. Answer it. Yes, answer it. Match column 1 with column 2. So it's simple. Griffith transformation, Hershey-Chase transduction, euchromatin active, prokaryotic DNA, it is nucleoid, right? It is nucleoid. So the correct answer will be this one. Second one. Next, replication is continuous in. Replication is continuous in. Replication, it is continuous in. The application is continuous in, of course, when it is the leading strand, right? So leading strand will form complementary to the template strand having polarity 3' to 5'. And even in your PCR polymerase chain reaction, right, the DNA replication is continuous. Clear? Okay, the next question, Hershey and Chase use which type of radioactive medium in the experiment? Hershey and Chase, they use which type of radioactive medium in the experiment? Quick. Quick. Hershey and Chase choose which type of radioactive medium in the experiment? Very simple it is. Very simple. Radioactive sulfur and radioactive phosphorus. Can you tell me the name also? S35 and P32. S35 and P32. Right? Next question. Which statement is not correct? Which statement is not correct? Which statement is incorrect? Quick. Again, DNA and RNA, are they able to mutate? Are they able to mutate? Tell me guys, how's the energy? How's the Josh? Right, we have solved around 44 questions. Right, 44 questions we have practiced. Right, in near about 30 minutes or less than 30 minutes, because initially I was just talking, hai na? So imagine in 30 minutes we have solved the 44 questions, and you know that here I am just repeating that question, I'm explaining the answer. So what we have to do, we have to make sure that in final NEET examination we finish our biology paper in 35 to 40 minutes maximum with its checking. Okay, so show me the some energy guys in the chat section, quick. Yes, I want to see some energy in the chat section, quick. Yes. Yeah, please tell me about your doubt as well, guys, and quickly hit the like button. And tomorrow, you know now, and yeah, make sure that you join this AIMT as well. And tomorrow, what do we have? We have our HSP search class, hai na? What do we have? We have our HSP search class. So make sure you hit the like button here, right? So here also what you have to do, notify me. Where is search class? Right, click on that notify me. Your HSP sir is going to take the class tomorrow and he'll be teaching you the complete class 11th organic chemistry. Arey yaar, just a minute. See this, okay? See this. So make sure you hit the like button, okay? So he will teach you organic chemistry tomorrow, okay? Chalo, so let's start. So which statement is not correct? Right, so DNA, RNA, are they able to mutate? RNA being unstable mutate at a faster rate. The presence of thymine at the place of uracil confers additional stability to DNA. DNA is not dependent on RNA for synthesis of protein. For synthesis of protein, not possible, because DNA will form mRNA, mRNA will form protein, so DNA is dependent. So fourth will be the correct answer. Now let's move to the next question. Okay, which of the above level includes in regulation of gene expression in eukaryotes? Right, so be very specific. They are asking about the eukaryotes. Okay, so at which level we can control the gene expression? Tell me. Tell me. Just a minute. Okay. So, what do you think? What should be the answer? This, right? All four. Now move to the next question. Match the column 1 with column 2. Match the column 1 with column 2. Quick, match the column 1 with column 2. So just practice all the question and trust me, right? In your final NEET paper, from molecular basis of inheritance question are going to come from this lecture only. Okay, so answer it, answer it, answer it. So Z will code for this. Y will code for this. A will code for this. I will code for this. So 1 with C. So we got the answer. Next question. Allelic sequence variation has traditionally been described as a DNA polymorphism if more than one allele at locus occurs in human population with a frequency. So again it is from principles of inheritance and variation, but I believe you can answer it. If the frequency is greater than anyone, if the frequency is greater than, right bache, if it is greater than, obviously greater than 0.01, not 0.1, 0.01. Clear bache? Clear, clear, clear. Next question. Which major approach of HGP used for identifying all the genes that are expressed as RNA? Right? Which major approach of HGP is used for identifying all the genes that are expressed as RNA? All the genes that are expressed as RNA. Obviously it will be EST, express sequence tagging. Okay, next question. Again the incorrect match. Okay, again the incorrect match. Quick, again the incorrect match. A very simple question, very simple question. So purine, what is the trick that we have? Puga, it's G and A, right? It's guanine and adenine, right? It's guanine and adenine. What is it? It is guanine and adenine. Is that clear? Is that clear? Right? So fourth will be the correct answer here. Next question. Give the correct sequence of Hershey and Chase experiment. Give the correct sequence of Hershey and Chase experiment. I'm sure you know the answer. Correct sequence of Hershey and Chase experiment. Yes, the first will be infection, blending, and then there will be the centrifugation. Understood? Then there will be the centrifugation. Next, if DNA contains sulfur instead of phosphorus and proteins contain phosphorus instead of sulfur, what would have been the outcome of Hershey and Chase experiment? Nice question, and I know you know the answer. I know you know the answer, read it. They specified that sulfur will be there in the DNA, and you know that it is the DNA that will pass to the next generation, right? So sulfur should be in the bacterial cells, है ना? Because phosphorus is the component of DNA, that's why it passed to the bacterial cell, to the right, from the virus it passes to the bacterial cell. So now if sulfur is the component of DNA, then sulfur will pass, okay, then sulfur will pass to the bacterial content from the virus. Next question. In lac operin, the Z gene codes for. In lac operin, the Z gene codes for. The Z gene codes for. Simple. Beta galactosidase. Next. Match the column type of question. So, bache, they all are PYQs, right? They all are PYQs. Don't you think that it is just same? Everything is as per NCERT? Yes, Priya? Don't you think that everything is as per NCERT? It is that simple. How bad you people are, ma'am. Move. No, I will not. I will not. No, ma'am, move. Yeah, I'm explaining something. I'm not wasting your time like Wasim sir. You know? I'm not wasting your time like Wasim sir. Move. No, I will not. I will not. I will not. So if someone will ask you who's the most sincere teacher on Unacademy NEET English, that is me, Ambika Sharma. That's what you have to remember. Who's the most annoying teacher? Your Wasim sir. Again, no, I'm not going to move. Ah, Baba is the most sincere. Acha, what about me? What about me? Even I am sincere, yaar. I am sincere. I know, I was expecting this answer that HSP Baba will be the most sincere teacher. But I am also the one, yaar. I am also the one. Chalo, ठीक है. Who's the cutest teacher on Unacademy NEET English? Who's the cutest teacher on Unacademy NEET English? Cutest teacher. I can bet Wasim sir can never be cute. He's not at all cute. Please. If you are saying he's the cutest teacher, sorry, you don't have good taste. He can never be cute. If he will shave all that beard and all, then I can think of it. Yeah, might be he's cute. Because we don't know the hidden face there. Even for the Yawar sir also the story is same. Okay? So if you are saying that he is the cutest one, sorry, I cannot. You don't have good taste. Even Yawar sir is not cute, yaar. Please. Chalo. So focus here. Haploid content, we know that. This. E. coli, this. So C with 4, D with 3. C with 4, D with 3. Okay. Then we have to cancel these two. Then bacteriophage, lambda and psi. So A will go with 2. Third is the answer. Next, statement-based question. Furkana, statement-based question. So answer it. Given below are two statements. Statement 1, DNA polymerase catalyze polymerization only in one direction, that is 5' to 3'. Okay, that is 5' to 3'. And statement 2 is saying that during replication of DNA on one strand the replication is continuous while on another strand it is discontinuous. So what should be the answer? Yes, so obviously both the statements are correct because polymerization will be in 5' to 3' direction always, right? The bases will be added like this and one strand is continuous, another is discontinuous. So solution is 1. So match the list with list 2. Again, it's a very simple question. I will code for repressor. Z will code for beta galactosidase. Y will code for permease. A will code for trans-acetylase. Is that clear? Is that clear? Okay, so solution 2. Next question. In 10 E. coli cells, okay, with 15N double-stranded DNA are incubated in a medium having 14N nucleotide. So after 60 minutes, what are you going to get? Good question. Dekho, 10 cells were there initially. So after 20 minutes it will be, after 20 minutes it will be 20 cells, है ना? 20 cells. But what are they asking? How many E. coli cells will have DNA totally free from 15N? Right? Totally free from 15N. Guys, it's very simple. They are asking totally free from 15 cells. So starting May, if they have 15N, right, you are shifting it in a medium where 14N is present. So that will be the scenario. Once that will be the scenario. Okay, that will be the scenario. Okay, okay, so this is having 15N isotope, this red one is showing 14N. So basically after one generation you are going to get the hybrids. So you cannot say that ki after 20 minutes if you are checking it, they'll be free from, right, ki they will be free from your, what do we call it, they are free from your 15N, not possible. So now when you talk about the next generation, see, with the help of only one DNA molecule you can understand it. It will be like this. It will be like this. So see this. So in next generation what will happen? The 2R hybrid, 2R 14N. Now 20 plus 20 40, 40 plus 20 means they are talking about the third generation now, right? So after it you have to consider the third generation. So second generation mein that's the scenario. Second generation mein that's the scenario. So when you will consider third generation, that will be the case. See this. On the basis of that you can calculate it. So you have to tell that how many cells are free from 15N. How many daughter cells? Yeah, how many cells are free from 15 cells? So you can just take the example of, one DNA and you can explain it. So dekho, after 20 minutes both are hybrid. After 40 minutes 2 are hybrid, 2 are 14N. After 60 minutes, right, what will be the scenario? 2 will be intermediate, 6 will be 14N. So that is what they are asking. That is what they are asking. They are asking the cells totally free from 15N. So calculate it, right? Calculate it. So 6 cells are totally free. So you can multiply it with the 10. You will get your solution. That is 60 cells. Got it or not? Right? Dekho, I picked up one DNA. You know that it is having 15N isotope of nitrogen. I shifted it into a medium where 14N is present. E. coli is having a generation time of 20 minutes. So after 20 minutes when I will check my daughter cells, they'll be the hybrid. So after next 20 minutes when I will check my daughter cells, 2 daughter cells are hybrid, 2 daughter cells are having 14N only. After third generation when I will check, so I have total 8, right? I have total 8 cells. So in that 8 cells, 2 are having the 15N, basically the hybrid, and 6 are basically 14N. They are free from 15N. So in the starting we have the 10 cells. So simply I multiplied it with 10. I got my answer. 60 cells are there. Okay, 60 cells are there. Clear, 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 clear. And one student is asking about this. कि मैम हाउ इज इट पॉसिबल कि सिंथेसिस इज इन, बच्चे पॉलीमराइजेशन देखो, पॉलीमराइजेशन मीन्स right in which direction your bases will be added, and I always told you that synthesis is always in 5' to 3' direction. Okay, that's the answer there. That's the answer there. So now let's move to the next question and tell me, it is a good question na? It is a good question na? It's a good question, right? Awesome, very good. Next question. If the length of the DNA is 1.1 meter, what will be the appropriate number of base pairs? Again very simple. Again very simple. Right, they are saying the length of the DNA is 1.1 meter. The length of the DNA is 1.1 meter. Exactly, right. Haploid, 2.2 meter means diploid, 1.1 meter means haploid. So fourth will be the correct answer. Next, in an E. coli strain, I gene get mutated and the product cannot bind the inducer. So if growth medium is provided with lactose, what will be the outcome? Yes, what will be the outcome? Take your time to answer this. Take your time to answer this. Quick. They are saying if in an E. coli strain, I gene is getting mutated and the product is not binding with the inducer. Dekho, very simple. If product is not binding with the inducer, right, so obviously it is attached to the operator only. It is not just getting attached with the inducer. So what is going to happen? Right, the switching on of the operon will not be there. Why? Because the product of I gene, that is your repressor, it interacts with the operator, right? It is attached to the operator. So if we want to start this operon, we have to make sure that this repressor should have affair with lactose. So basically, you remember that story, that breakup story that I told you? Do you remember, Priya, that breakup story that I told you? That what is going to happen here? What is going to happen here, students? It's very simple. It's very simple. The point is, ki, if we want the transcription to start here, we don't want repressor to get attached to the operator. So basically, we want their breakup. So we will introduce our inducer, right? That is lactose. So lactose will attract this repressor. They both will pair up, and the site for the operator, the operator will be free. So RNA polymerase will come, that will bind to your promoter. So that's how it works. Clear, bache? That's how it works. So if it is not getting attached to the inducer, then simply second will be the answer. Clear? Chalo, next question. If a geneticist uses the blind approach of sequencing the whole genome, we have already solved it, right? When it is the whole genome, sequence annotation will be there. Okay, chalo. The process of translation of mRNA to protein begins as soon as it will begin as soon as, tricky one? Nahin. Of course, the fourth one. Remember, smaller subunit, it will get attached to the mRNA. So if you are talking about the prokaryotes will be having the Shine-Dalgarno sequence in the mRNA, and 30S will be having anti-Shine-Dalgarno sequence. Next question. DNA polymorphism forms the basis of. I still remember this question, right? Do you know this is a question because of that, the 720 out of 720 was not there. Last to last year. This was the question. Okay, this was the question. So the correct answer is second one, right? Both genetic mapping and DNA fingerprinting, isn't it? What should be the answer? DNA polymorphism forms the basis of. It forms the basis of, exactly, both. So many, even teachers, they marked it incorrectly. Then read the following statements and choose the correct option. Do it quickly, then we will. Okay, so the next chapter, the question, just a minute. Chalo, wait. It's lagging. Just a minute. It's lagging. Till then you can talk to me. Okay, fine. Haan bache, soon we are going to start the questions from the next chapter. Yes. After 5 to 6 questions, I'll be asking the questions from the next chapter. Don't worry about it. Okay? Don't worry about it. So read the following statement and choose the set of correct statements. What should be the answer? What should be the answer here? Quick. Euchromatin is loosely packed? Yes. Heterochromatin is transcriptionally active? No. right? So wherever there is B, it is not our correct answer. Then histone is wrapped by negatively charged DNA, yes. Histones are rich in lysine and arginine, yes. Typical nucleosome contain 400 base pairs, no, it should be 200. So we got our answer, that is 1. Okay, that is 1. Next, what is the polarity of continuously synthesized new strands of DNA? Polarity of continuously synthesized new strand of DNA. They are telling us continuously synthesized. Of course, 5' to 3', and synthesis is always in 5' to 3'. Next, which type of RNA polymerase is responsible for tRNA and 5S or ribosomal RNA synthesis? Of course, RNA polymerase 3. Here also the answer is 1. Next, which factor serves as the initiation factor to initiate the process of transcription? To initiate the process of transcription, again 1 is the correct answer, sigma factor. Next, for initiation of translation, the ribosome binds to the mRNA at the start codon, that is recognized by the initiated tRNA. That start codon is, again answer is 1, AUG. Next, label A and B correctly. Dekha, I told you we can ask, we can get such questions. What is A, what is B? What is A, what is B? Again, A 5' end because it is having free phosphate. B 3' end, free OH. Again 1. Next, which statement written below states the points which confer stability of helical structure of DNA? It is as such NCERT. It is as such NCERT. Yes, it is as such NCERT. Quick. Exactly, the plane of one base pair stacks over the another. And yes, hydrogen bonding between. Which statement written below states the points which confer stability? Okay, it is not just about the additional stability, total stability. Dekha, even I marked it incorrectly. Both. Third is the answer. Third is the answer. So that is why sometimes we need to read all the statements, right? Even I marked it incorrectly. So here both the statements. Next. Okay, so in the given figure, identify A and B. Identify A and B. Identify A and B. What should be the correct answer here, Pujitha? Quick. What should be the correct answer? What should be the correct answer? It is third. RNA polymerase sigma. Okay. One second. Not this one, because see, RNA is ready. So yes, A will be the RNA polymerase and B will be your raw factor. है ना? B will be your raw factor. And the process is termination. Next, if the sequence of bases in DNA template is this, what will be the sequence of bases in the transcript? Good question. Template is having this. What will be there on the transcript? Good question. Good question. Quick. I know you know the answer, guys. Speed up. Exactly, the second will be the correct answer here. Next, bache, and make sure whenever you are making the RNA, right, at the place of thymine, just A will form bond with T, but from DNA you are making the RNA, so here it will be U, then AA like this. So second is the correct answer. Now next one, name the component that binds to the operator region of an operon and prevents RNA polymerase from transcribing the operon. Name the component that binds to the operator. Obviously it is the repressor. It is the repressor. Next, unequivocal proof. I don't think that we need to solve it. We know it. EST, all the genes, no, that are expressed as proteins, है ना? That are expressed as proteins or RNA. EST में क्या आएगा? What will be there in EST? 1 or 4? 1 or 4? Exactly, it should be all the genes that are expressed as RNA. Next, what is the role of RNA polymerase 3 in the process of transcription? Role of RNA polymerase 3 in the process of transcription. You know the answer. Again, transcribes this tRNA, msRNA and snRNA. Is that clear? Is that clear? Chaliye. So now let's move to the next chapter. Right, we have covered everything from this part. So, chalo, answer this question. Which of the following RNA is not required for synthesis of protein? Not required for synthesis of protein. Yes, not required for synthesis of protein. Exactly, small interfering RNA. Next question. The separated fragments resulted from treatment with a restriction enzyme have an overhanging piece of single strand DNA at the ends. They are known as overhangs, obviously the sticky ends. So can you just give me one example of an enzyme which produces sticky ends? Only one example I'm asking, only one example that will give you sticky ends. Only one example that will give you the sticky ends. Exactly, that will be EcoR1. Those who are writing the ligase, please check my lecture. Okay, next, DNA polymerase, Taq polymerase used in PCR is isolated from, it is isolated from, bache, see, yeah, Hind 3 is also going to give you the sticky end. Desati, very good, very good. So it should be bacteria, archaebacteria. Next, identify A, B, C and D. Dekha, I told you, you have to remember it, question can come. What is A, what is B, what is C, what is D? Hanji. It's simple, trust me, it's simple. A and B. What should be the answer? Exactly, you all are right. The answer is going to be fourth. Right, bache? Clear? Next question. Which vector can clone only a small fragment of DNA? The word is small fragment now. Right? The word is small fragment now. The word is small fragment now. The word is small fragment now. That is plasmid. Okay, that is plasmid. Next question. After the formation of products in the bioreactor, it undergoes through some processes before a finished product is ready for the marketing. That will be downstream processing. Next question. What is the source of TI plasmid which is modified and used as a cloning vector to deliver the desirable gene into the plant cell? What is the source of TI plasmid? Exactly, Agrobacterium tumefaciens, also known as natural engineer, and it's an engineer without the degree of engineering. Okay? So it's a soil-borne bacteria. It's a soil-borne bacteria. Next, purified DNA ultimately precipitate out after the addition of chilled ethanol. The DNA that separates out can be removed by, I'm pretty sure my students know the answer. Acha Kapil, thank you. But I don't think so, Kapil. Exactly, that will be spooling. Understood? Next question. Okay, in the following figure, which DNA is not digested? Which DNA is not digested? Not digested. Which DNA is not digested? Yes, you are right, the one will be the correct answer. Dekho, rest all are, they are having the fragments that are moving. So this one is not digested, right? Only one fragment which is present here. Next, PBR322 has two antibiotic resistant genes. They are, it is the easiest question. They are ampicillin resistant, tetracycline resistant, right? Next question. The construction of first rDNA was done by, we have even discussed that, the construction of first rDNA was done by Stanley Cohen and Herbert Boyer. Understood? Next, alternative selectable markers developed to differentiate non-recombinants from recombinants on the basis of. Alternative selectable markers which are developed to differentiate non-recombinants from the recombinants are on the basis of exactly color producing, chromogenic. Remember? Next, which enzyme is used in PCR? 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 Thermostable DNA polymerase, your TAC polymerase. Your TAC polymerase. Next, the definition biotechnology is given by EFB for the definition biotechnology is given by EFB. Guys, you have to solve as much questions as you can. So quickly answer it. So answer will be 3. Right? Next question. Dash gene codes for the proteins required for replication of plasmid. Right? Required for the replication of plasmid. Required for replication of plasmid. It is ROP, right? Next question. First recombinant DNA was made by linking dash with a dash and dash. Yes. It is also simple. Please read it carefully, people. Please read it carefully. Yes. So antibiotic resistant gene, yes. So it is 1. Why 1? Because it was of Salmonella typhimurium. Right? That plasmid was of Salmonella typhimurium. Next question. Eco R1. R is derived from. Easiest question ever. Trust me. Easiest question ever. Quick. Strain, right? Next, restriction endonucleases cut which bonds? They'll cut which bonds? The endonuclease. They'll cut which bonds? Be sincere, bache. They will break sugar phosphate backbone, means phosphodiester bond. Okay, phosphodiester bond. Next, the separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as, yes people, quickly hit the like button as well, and make sure you watch the lecture that I have conducted yesterday for your quick revision. Yes. So what should be the answer here? Illusion. Next, recombinant host cell in which foreign DNA is present in the coding sequence of the enzyme beta-galactosidase will produce. Right, recombinant host cell in which foreign DNA is present in the coding sequence of enzyme beta-galactosidase will produce. It will produce. You have beta-galactosidase. Then there will be, read it carefully. They are saying recombinant host cell in which foreign DNA is present in the coding sequence of beta-galactosidase. If beta-galactosidase is present, blue colored colonies will be there. But if within that sequence a foreign gene is introduced, so insertional inactivation can occur and white colored colonies will be there. Okay, white colored colonies will be there. So complete the analogy with respect to the DNA isolation. Bacteria, lysosome, हाँ जी, what should be there? Fungus with, जैसे DNA isolation में bacteria, lysosome and fungus with chitinase. Why? Because the cell wall of fungal contain chitin. So what is chitin? It's a homopolymer. It is a homopolymer of NAG, N-acetyl glucosamine. Next, to make the host cell competent, they had treated with specific concentration of They are treated with specific concentration of, remember, torture, Pallavi. Yes, Akanksha, Pallavi, Preeti. Remember that torture. What should be the answer? Calcium ion. Calcium ion. Next, select the correct sequence for steps involved in PCR. Select the correct sequence for the steps involved in PCR. It's denaturation, annealing and extension. Next, downstream processing is simple: separation and purification of product. Next, if DNA is inserted within the coding sequence of beta-galactosidase enzyme, then, right, insertional inactivation will occur. Insertional inactivation will occur. Yes. Then obviously what is going to happen? It's a very simple question. It's a very simple question. As of now, we have practiced 104 questions. So tell me your score as well. Right? Tell me your score as well. Guys, it's very simple. They are saying if you have inserted DNA within the coding sequence, then non-recombinants do not produce color. No, bache. It's not like that. Non-recombinants will give blue colored colonies. ठीक है. Recombinant will give blue colored colonies. No. So A1 should be the answer now. Non-recombinant, because देखो ना, non-recombinant means non-transformant. So if it is non-transformant, beta-galactosidase expression will be there and that will give the blue color. So tell me your score out of 104. Anji, tell me. We have solved 104 questions in 1 hour. Hanji, 100 are correct. Okay. Very good. So how many questions are correct? At least tell me that. 95 are correct. No. You need to work hard then. I can expect ki haan theek hai ma'am, 102 are correct, 100 are correct, but not less than that, yaar. Not less than that. Literally, not less than that. 100 is still fine. 100 is still fine because whatever I am asking today, we have learned it yesterday. We have revised it. Okay, we have revised it. Chalo, now move to the next question. If the plasmid in bacteria does not have any insert, then the colony is produced. Plasmid is not having any insert. Exactly, the solution is 1. Next, identify the correct match from column 1, 2 and 3. From column 1, 2 and 3. From all the 3 columns you have to identify the correct match. Very simple. It's very simple. Exactly, the 1 will be the correct answer. Selectable marker. Yes, ampicillin resistant gene is selectable marker, help in the identification of recombinants. Right, here restriction in endonuclease is not going to cut randomly. It's specific. It's not linear DNA, it's circular one. BAC, host cell. Isolation, no, it will help in the cloning. So 1 should be the correct answer. So method in which alien DNA is directly injected into the nucleus. You are directly injecting it into the nucleus. Directly we are injecting it into the nucleus. Quick. Exactly, micro injection. Next, which of the following is not a feature of the cloning vector? Not a feature of the cloning vector. Not the cloning. Exactly, second, many restriction sites of one restriction enzyme. Nahi. Cloning vector can have many restriction sites of more than one restriction enzyme. Okay, next, gel electrophoresis is used for. Gel electrophoresis is used for. Gel electrophoresis is used for. Separation of DNA fragment as per their size, not as per their charge. Next, insertional inactivation is used for insertional inactivation is used for selection of transformed cells. Next, selection of Hanji. So don't you think it's kind of same? Insertional inactivation is used for recombinants, and don't you think we can consider it as the transformed cells also? Transformed cells also. So you know na, two options are correct. Two options are correct. So you will get the bonus marks if out of 1 and 4 you have answered it. Remember, I posted this video. I don't know whether you have seen it or not, for the bonus marks. Ye dekho. This screen is having some issue with me. I'm quite sure about it. See this. For the bonus marks. You can check it out. Okay. Next. The construction of first rDNA emerged from the possibility of linking a gene encoding antibiotic resistant gene with native plasmid of. The plasmid was of exactly Salmonella typhimurium. Next, for a DNA to function as a cloning vector, the most essential requirement is exactly origin of replication. Next, among the following, select which is not the tool of rDNA technology. Among the following, select which is not the tool of rDNA technology. Which is not the tool of rDNA technology. Not the tool of rDNA technology. Tool, of course. Promoter. Next, Taq polymerase is used in PCR because of its thermostability. It is active during high temperature. Next, which one is not a basic step in genetically modifying an organism? Not a basic step. Not a basic step. Correct. Next, which one of the following is must in biotechnology? Bache, they are asking about must in biotechnology. So yes, we need these two things. But even in in the tools, alkaline phosphatase are also there. I won't say that is the must, but yeah, these two enzymes are the most important one. One will cut the DNA at specific sequence, and one will join the sugar phosphate backbone. Next, select the incorrect statement. Read it. Select the incorrect statement. Tell me. You have to select the incorrect one. What should be the answer? Very good. It will be C, D and E, right? C, D and E. Next, which of the following is used to deliver desirable gene in the animal cell? In the animal cell. Animal cell. For the animal cell, what do we use? For the animal cell, what do we use? Disarmed retrovirus. Disarmed retrovirus. So core technique of biotech, which involve maintenance of sterile ambiences, which involves maintenance of sterile ambience is, yes, which involves maintenance of sterile ambience is, of course, it is the bioprocess engineering. Okay, bioprocess engineering. Next, genetic engineering is better over traditional hybridization. What should be the answer? What should be the answer? What should be the answer? Exactly, introducing the desirable genes only. Introducing the desirable genes only. Next, restriction endonuclease and methylase were isolated in E. coli. They are asking about the restriction modification, right? They are asking about the restriction modification system. So it was isolated in which year? We discussed it yesterday. Exactly, it will be 1963, right? So now guys, how's the energy? How's the Josh? How's your confidence level? How's your confidence level? Nandini, Nikita, Chetu, yes, bache kaisa, Matya, Monisha, VN. How's the confidence level? Quick. Show some energy in the chat section. At least I should know now. How are you feeling? Quick. How's the energy? Imagine all the questions from this session will be there in your NEET 2024. You know the answer of all the questions. Imagine you know the answer of all the questions there, right? You are getting in biology 360 out of 360. In chemistry you are getting 130. In physics you are getting 180. Oh my god. Chalo, chemistry maybe let's keep it 180. Then oh my god, dream come true, ma'am. है ना, dream come true. है ना मैम, oh my god, then तो मैम you can ask us anything. Chalo, so let's move to the next question. Choose the odd one out with respect to the ends produced by the eco R1 sticky ends. Eco R1 is going to recognize this sequence. It is gatus. It is gatus. Understood? Next. Oh. Seriously. Odd one out. I'm sorry. My bad. Odd one out. Dekha? Odd one out. You know, even I started imagining that you are getting 720 out of 720. My bad. So yes, odd one out they are asking. So it will be blunt ends. So what can you learn from all this? That even teachers can make the mistake when they are too excited. So whenever you answer your questions in your final paper, make sure that you are calm, right? You are composed and you are reading all the options. Chalo, next. The separated DNA fragments can be visualized only after staining the DNA, and then there are exposure by, obviously it is the ethidium bromide, and then UV radiations. Next, if we ligate foreign DNA at BAMH1 site of PBR322, then what is going to happen? Where is that BAMH1? Dekho, it is very important. Yes, you have to remember. You need to remember the PBR322, right? You need to remember the PBR322 diagram. It's very, very, very important. So promise me by tomorrow you will complete it. Tell me, promise me by tomorrow you will complete it. By tomorrow you will complete it. Promise me that. Yup. Okay, so what should be the answer here? What should be the answer here? So exactly the answer is 1. Okay, the answer is 1. I don't think that I need to explain it. So if there is the resistance to ampicillin, so it means there will be the sensitivity to tetracycline, because right, BAMH1, it is located, the restriction site for it is located in that tetracycline gene. So tetracycline gene. So when we will use this restriction enzyme, the tetracycline will become inactive. This is the insertional inactivation. Next, which vector is suitable to deliver the desirable gene into the dicot plant? Which vector is suitable to deliver the desirable gene into the dicot plant? Quick! Which vector is suitable to deliver the desirable gene into dicot plants? Dicot plants, dicot plants, dicot plants. So it will be disarmed TI plasmid of Agrobacterium. Next question. All the following are the methods of direct gene transfer except direct gene transfer, which is mainly in the animal cell, except disarmed pathogen. Okay, next. Which of the following is incorrect with respect to PCR? Incorrect. Incorrect. Quick, quick, quick. Incorrect. Exactly. Single set of RNA primer. It is incorrect. What is not present in the simple stir tank bioreactor? Not present. The word is not present because it is simple stir tank bioreactor. Obviously, a sparger will not be there. Which Roman numbers following the names of restriction endonuclease indicate? Yes, it indicates. What are they going to indicate? Read it carefully. Yes. So the order in which enzymes were isolated from the strain of bacteria. Next, in PCR technology, primer is a. Okay, in PCR technology, primer is a. What is a primer? In PCR technology, the primer is a. In PCR technology, primer is a. So it's a oligonucleotide, isn't it? Isn't it? Complementary. It is not identical. Identical means same. No. No, it will be complementary to the region of DNA. That's why 1 is the correct answer. Next, thermostable DNA polymerase used in PCR is isolated from? It is isolated from very simple Thermus aquaticus. Very simple Thermus aquaticus. Next, ligation of foreign DNA at which of the following site will result in the loss of tetracycline? Dekha, this is a repeated question. Tetracycline resistant. Tetracycline resistant. Quick. Tetracycline resistant. Obviously the answer is 4, right? So in tetracycline you have this. At least remember the from PBR 322 that where the insertional inactivation can occur. At least you need to remember that. Okay. Chalo, next question. Which of the following statement is incorrect about Agrobacterium tumefaciens? Which of the following statement is incorrect about Agrobacterium tumefaciens? Quick. Incorrect about the Agrobacterium tumefaciens. Quick. Incorrect about the Agrobacterium tumefaciens. देखो, these two are for sure correct. Now it is used to deliver gene of interest in both prokaryotic and eukaryotic host cell. If you remember, I told you TI plasmid is a shuttle vector. This is what I have explained you, right? So TI plasmid from Agrobacterium used for gene transfer is not pathogenic to plant cell. So TI plasmid, is it not pathogenic to plant cell? You have to figure out the incorrect one. So don't you think second TI plasmid from Agrobacterium used for, अच्छा, which is not used for, okay, which is used for gene transfer. देखो, नहीं students, check it again. You have to tell the incorrect statement. Which statement you need to answer? You need to answer the incorrect statement. देखो, incorrect statement. So what will be that incorrect statement? Try to answer it. Incorrect statement. They are saying कि TI plasmid from Agrobacterium tumefaciens, which you are using for gene transfer, it will be not pathogenic because we have already removed the tDNA. I need to check this option. Because TI plasmid in general it is pathogenic, but we are using it for gene transfer, so I have to check it. Okay, I have to check it. I'll tell you about it. So right now you can put a question mark over it. I'll tell you. Chalo, next question. So yes, Hanji, match the following. Quick. What should be the answer? Just a minute. Chalo, let's go. So kanamycin, disarmed retrovirus, C will go with 1. Okay, so you can cancel these two option. So kanamycin, right, it will be the selectable marker. This clay one will be your restrictions. This will be the selectable marker, D with 2. So do we have, yes, third will be the correct answer, right? Third will be the correct answer, isn't it? 3 will, how is it possible? Guys, 1 second. A is antibiotic resistant. Kanamycin is an antibiotic. है ना? Just a minute. Let me check the answer. It's very simple. Kanamycin R gene. It's a selectable marker. Yes or no? It's a selectable marker, and it will provide the resistance against the antibiotic. Okay, it will act like a selectable marker and it will provide the resistance against the antibiotic. Okay, fine. D with 4. Yeah, it will be better. D with 4. Okay. Disarmed pathogen will deliver it into the 1. So C with 1. Fine. So Claire 1, it meets the restriction sites, and kanamycin, yes, this antibiotic will be the selectable marker. Okay, fine. ठीक है बच्चे, so 1 is the correct answer here. 1 is the correct answer here. Right बच्चे? Right. चलो next. Which of the following can act as a molecular Caesar? I think it is the easiest question. Restriction enzyme. Next, during the purification process for rDNA technology, addition of chilled ethanol precipitates out exactly the DNA. Chilled ethanol will precipitate out the DNA. Next question, in gene gun method, which is used to introduce alien DNA into the host cell, microparticles of dash metal are used. Right? Microparticles of tungsten or gold, they are used. Clear bache? Tungsten or gold, they are used. Clear bache? So upon exposure to UV radiation, DNA stained with ethidium bromide will give you which color bands? Bright orange color. So it is again the favorite question of your examiner. Next, main steps in the formation of rDNA. So it's a NEET 2023 question. You know na, 2 options were correct here. No doubt they marked this as solution. 4th, ki B first, then C, then D, then A. But if you remember, right, grace marks were there for this particular question. Do you remember that? Grace marks were there for this particular question. Okay, so again, bonus marks were provided. It's a NEET 2023 question. Next, which of the following is not a cloning vector? Of course, probe. Right? It is radio-labeled DNA, single-stranded DNA or RNA. It is not a cloning vector. Next, in the following palindromic base sequences of DNA, which one can be cut easily by a particular restriction enzyme? Right? Palindromic base sequence. They are saying palindromic base sequence. Hanji, what should be the answer? Exactly 1, and that is for EcoR1. Next, restriction into nuclease recognize specific sequence to cut DNA known as palindromic nucleotide sequence. Statement 2 is saying restriction into nucleases cuts the DNA strand a little away from the center of the palindromic sequence. What should be the answer? What should be the answer? Exactly, right? Both the statements are correct. Next, which of the following is not a desirable feature of the cloning vector? Not a desirable feature of the cloning vector. Not desirable. Hanji, not desirable. Exactly. Presence of 2 or more recognition site, it is not desirable. It is present, but it is not desirable. Next, plasmid PBR322. So dekha, many questions are there from PBR322. So make sure you remember that diagram. That's important. Right, so it is having PST1 restriction, enzyme site within gene amplification for gene ampicillin resistant that confers ampicillin resistance. So in this enzyme is used for inserting a gene for beta galactosidase production and recombinant plasmid. So what should be the answer here? Yes, exactly. The 1 is the answer because ampicillin resistance will not be there to that particular host cell then, right bache? Next, specific recognition sequence identified by endonuclease to make cuts at specific position. Do we need to repeat it? No ma'am. No ma'am. Hai na? No ma'am. Chalo. So answer this question. Which of the following technique is helpful in genetically engineered living organism? Of course, it is the rDNA technique, right? Chalo. So next is from, so bache, only few questions are left and then we will start your next chapter. Okay? Then we will start your next chapter after few questions. Okay, and the next chapter that we are going to start is It is. Chalo, we'll be starting it soon. The next chapter. Right? Chalo, so biological process of RNA can silence specific genes using, what will they use? What will they use? Acha, you need a small break. After 2 hours, right? I just started the class at 10:10. So at 12:10 I'll give you the break. Don't worry. So what should be the correct answer here? 1. है ना? So next, the amount of protein, human alpha lactalbumin present in the milk produced by first transgenic cow. It's important. It's important. What should be the answer? What should be the answer? It's a very simple thing, directly from NCERT. What should be the answer? I want all of you to give the answer. Quick, students. What should be the answer? It's very simple. It's second. Now, the stirrer in the simple stirred tank bioreactor facilitate. The stirrer in the simple stirs tank bioreactor will facilitate. It will facilitate. Yes, availability of oxygen throughout the bioreactor. That's the function of stirrer, the uniform availability of the oxygen throughout that bioreactor. Okay, so which type of vector is used to introduce a functional ADA cDNA into the lymphocyte during gene therapy? Right, that will be a very good, Deepshikha. Yes, very good. It will be disarmed retrovirus. What is the meaning of disarmed retrovirus? The meaning of disarmed retrovirus is it doesn't have the harmful genes. Okay, it doesn't have the harmful genes. Next question, for chemical safety testing, transgenic animals are made which are yes, which are, you need to figure out the correct statement. Which are exactly right. Right, what is the correct answer? We have to make the transgenic animals that are more sensitive to toxic substance so that we can check how that drug is affecting them. Okay, so not correctly matched. This one correct. This one correct. This one correct. This is not correct. Second answer. Next, consider the following statement with respect to the gel electrophoresis and choose the correct option. DNA fragments move from cathode to anode. The most commonly used matrix is agarose, which is extracted from seaweed. So we know the answer, right? We know the answer. Very good, Akanksha. So it's both A and B. Next, which of the following is used in the commercial production of human insulin? Commercial production of human insulin. So the correct answer here is, yes, it will be your E. coli. Then select the incorrect match. Select the incorrect match. Select the incorrect match. Exactly, the correct answer is your third one. Next, which among the following is true for the restriction endonuclease? Which among the following statement is true for restriction endonuclease? True, true, true. Are they present? They are used in vivo? No, it should be in vitro. They are present in mammalian cells? No. They can cut anywhere? No. They are synthesized by bacteria as a part of their defense? Correct. Right? So genes encoding resistance to antibiotics help in identifying and eliminating non-transformant and selectively permeating growth of the transformant. These antibiotic resistant gene in the vectors, they are considered as they are considered as very simple, they'll be considered as the selectable marker. देखो बच्चे, whatever questions we have solved, from that question what you people can conclude? Tell me, what you guys can conclude? Hanji, what you people can conclude? Even in the molecular basis of inheritance, they were all PYQs. Even in the biotechnology, they are all PYQs. You will see that questions are repeated in one way or another way. Same type of questions are repeated in one way or another way. है ना? Same type of questions are repeated. Everything is from NCERT. So if you solve last 10 years PYQs, if you will solve last 10 years PYQs, trust me on that, bache, you will get 330 plus marks in biology, right? For sure you will get it. For sure you will get it directly from the PYQs. Okay, so last 10 years, okay? So and if you can make it 15, that will be very good, but at least last 10 years. At least last 10 years. So significance of heat shock method in bacterial transformation is to facilitate what should be the answer? What should be the answer? Yes, Deepshika, very good. Hanji, Anna, what should be the answer? Obviously it is going to be 2, right? The DNA will enter through the pores that are created, right? Because the treatment of that ion, that divalent cation, it has created the pores now, so that will be the answer. So read the following statement and choose the correct option which correctly fills the blanks. Read the following statement and choose the correct option which correctly fills the blanks. Quick. To obtain a recombinant protein, the transformed cells can be multiplied in a continuous culture system wherein the used medium is drained out from one side, fresh medium is added, so that cells will be in their log phase, that is exponential phase, that is they are actively dividing. Clear? Next question. Complete the analogy. E. coli, EcoR1, Haemophilus influenzae, very simple. Third. Okay? Done. So answer this question, give it a try. A gene gun is used to shoot high velocity microparticles of dash coated with DNA into plant protoplast. Okay, so during heat shock method, bacterial cells are incubated with recombinant DNA on ice, followed by placing them at a temperature of. So this statement will give you the answer directly. It will give you the answer directly. Obviously here it should be 42 degrees Celsius, that's why our answer will be fourth. Okay. So a gene is expressed in a host organism which doesn't naturally have this gene. Such host organism is called as, again it is NCERT people, again it is NCERT. After it we are starting human health and disease. Okay, after this chapter we'll be starting human health and disease. The sequence will be same like we have done yesterday. Okay, so what should be the answer? It will be heterologous host. Okay, the second. Next, in the E. coli cloning vector PBR2, ROP codes for proteins involved in the replication of the plasmid. Clear, bache? Chalo. So answer this question. Proteins encoded by genes CRY1AB. It will be for the be quick. Hanji, Chetu, Nandini, Shaina, Hannah, we are moving very quickly. So today you will be free early. And then later on after this class, what are you going to do? You are going to practice, you are going to revise the topics that we have covered yesterday. Hannah, you are going to revise the topics that we have covered yesterday. Okay? Okay, chalo, what should be the correct answer here? Exactly, the corn borer. Next, insulin is synthesized as in mammals, insulin is synthesized as Insulin is synthesized as a, देखा beauty of biology. You have to read more, but you have to answer very quickly. Okay. So in mammals, including humans, insulin is synthesized as prohormone, means inactive hormone. ठीक है? चलिए. So same type of questions are there, and this is also we have practiced. Okay, so what might be the major advantage of beginning gene therapy prior to birth? Yes, prior to birth. Quick, guys, quick. Yes, because बच्चे, it will be a permanent cure. It will be a permanent cure. Okay, so what is granted by government to the inventor to prevent others from commercial use of his or her invention? है ना, I didn't leave even the NCERT line, not even a single NCERT line. So I think I deserve some likes. I deserve some likes, and this is what I'm expecting from you people. And make sure you check it also, the list of prohibited or the banned items that are not allowed, right? That are not allowed in your examination hall. So make sure you enroll for the mock test. Why am I telling you this? It's very simple, bachcho. Dekho, see, if you will give paper, it is from 2 p.m. to 5:20 p.m. Means for a month, because it is going to start from 31st March to 28th April. What is going to happen because of that? Right? You will be very clear. Right? You will be very clear ki yaar, your brain will be like from 2 p.m. to 5:20 p.m. I have to give the exam at any cost. I'll tell you, bache, I strongly believe in 80-20 rule, right? That your 80% of the paper will come from the 20% of the syllabus, okay? And you know that most of the syllabus, even if you see the YouTube videos randomly, you get something in your brain, okay? And when you are active, when your brain is active, when you are alert, you can manage everything. I'll give you one example. You all know it very well ki whenever we have the exam, even if we don't practice the writing, whenever we know, in the school also, this is the best case that I can explain right now, ki initially we start writing very slowly, but when we realize ki yeah, 2 hours are over, only 1 hour is left, then we start writing like this adrenaline rush. Just let's say a dog start following you, the dog is about to bite you. What will you do? You will run very fast. You will run very fast. You will be like, yaar nahi, I have to protect myself. Hai na? Why? Adrenaline rush. Your body is like, right now I have to survive in this situation. It is all about the survival. So every day when you will sit from 2 to 5:20, you will have that habit ki right, this is the time when I have to be more focused. Hai na? You will be more focused. You will be like, nahi yaar, no matter what, I have to write the exam from 2 to 5:20 p.m. So it will be your habit. Your brain will be alert, right? You are training your brain to be active at that particular time, right? You will not feel sleepy. Okay, you will not feel sleepy, and that's the problem with some students also, right? Some students, they feel sleepy at that time, they feel lame at that time, but you have to be very active. Okay, so from this particular month, from the April month itself, start making it a habit कि I have to tone my body in this way, that I am going to relax in that morning period. I need to be very active in this time period. So please make sure that you enroll here in AIMT. Make sure you enroll here in AIMT. I want to see maximum students here, and we are not asking for even a single rupee here. We are not even asking for a single rupee. Okay, so analyze the exam, and I always say whenever you get something something good to read, right, some exam to give, and that too is free of cost, give all the exams, right, give all the exams, sab bator lo. Okay, okay, chalo, come to this. So obviously it is the patent right, it is the patent right. Okay, chalo. So now answer this question. Answer this question. If we digest a linear DNA with a restriction enzyme having 4 recognition sites, it will form how many fragments? I'm pretty sure you know the answer. I'm pretty sure you know the answer. You answer it. I'll be back. Yes. Uh-oh. Uh-oh. Yes, there go. It's a linear DNA fragment. You have 4 sites. 1, 2, 3, 4. So you are making cut here, then here, 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 and then it's a linear fragment, no? Here, total 5 fragments will be there. Total 5 fragments will be there. Next question. Answer this question. Then we will move to human health and disease. We will move to human health and disease. Quick. Then we will move to human health and disease. Yes, they go alpha-1 antitrypsin to treat emphysema, retroviral vector, right. What will it do? Where will it be used? In gene therapy, ELISA, antigen antibody interaction, PCR amplification. So, Hanji, so what is the answer? This, alpha A is with 2. Okay, so you can mark it, I have already told you. Next question, which of the following is the name of transgenic sheep? Not the cow, the sheep. The sheep. What is the name of the sheep? What is the name of the sheep? Sheep. Dolly, sorry. Tracy darling, sheep. Is that Tracy darling? Tracy darling or Dolly darling? What's that? Name of the sheep, yeah, Tracy darling or Dolly darling? Name of the transgenic sheep, Tracy or Dolly? Dolly darling. Nahi, first hai na, first. Transgenic sheep. The word is transgenic sheep. Hanji, tell me. I think it's the Dolly darling, nahi? First one is Dolly darling. Ki Tracy? Tracy. That is Tracy darling. I like the name. So, nahi yaar, Tracy is the answer. Tracy darling is the answer. Nahi yaar, Tracy darling. Tracy or Dolly? Matlab now we have to fight for Tracy and Dolly. Wah yaar. Wah. Let's fight for Tracy and Dolly. No, Tracy darling. The answer is Tracy darling. ठीक है, चलो. Now, same type of questions are repeated. Aha. Transgenic models for the study of disease does not exist for. Transgenic models for the study of disease does not exist for. Does not exist for. Obviously, COVID-19. है ना? It's fun to solve the questions. Now, all the NCERT lines are almost completed. Done. Answer this question. Answer this question. I'm waiting for the human health and disease. We are about to reach there. So answer it, answer it, answer it, people. Answer it. Answer it. Quick. Identify A, B, C and D in the given diagram and choose the correct option. What is A? What is B? What is C? What is D? Quick, quick, quick. Quick. What should be the correct answer? Dekho, first of all, they both should be same. So A is not at all. 3 is not the answer. Okay, so it will be eco R1 only. Second, next. GM plants have been useful in many ways except. Hanji, except. Except, exactly. They'll have increased efficiency of the mineral usage. Okay. Bt toxin kills certain insects such as tobacco budworm, armyworm, etc. These insects belongs to which order? These insects belong to which order? Your tobacco budworm, armyworm. Worm. Very simple, very simple, very simple. Yeah, all of you please answer it. All of you please answer it. Again NCERT, exactly, the solution is second. The solution is second. How many applications given in the box are the applications of biotechnology? How many of them are the applications of biotechnology? How many of them are the applications of biotechnology? Arey, oh my god, the Mr. Skeezer is so upset. You are moving so fast. So what do you want me to do? Do we need to go slow? Ali mila bacha, why are you getting worried? Do we need to go slow? What do you want me to do? It's not the formality. Bacha, if I will go slow, then you people will start crying. And obviously, questions, so you have to answer like a flash because already we have covered it. Hana? Otherwise it will be mole. Otherwise it will be that na moe. Hana? Insta generation moe. Acha, I need to go slow. Okay. How many applications given in the box below are the applications of biotechnology? Are the application of biotechnology? All are the applications of biotechnology. 1, 2, 3, 4. They all are the application of biotechnology. Okay? Okay. Can I move now? Can I move to the next question now? Okay, so Eli Lilly, an American company, they prepared 2 DNA sequence A and B of human insulin and introduced them into the plasmid. What should be the correct option? Acha, aise nahi, normal padhao? Acha, normal padhao? Okay. So what should be the correct answer here? Be quick. What should be the correct answer here? Yaar, why are you getting worried? Ma'am, you are moving too fast. The same questions are there, yaar. Chill. So obviously our favorite, one and only, Mr. E. coli, है ना, मिस्टर E. coli. Now ना I'm bored with this topic. ठीक है, so now let's move to the next question. Let's move to the next chapter. All these questions are okay. Identify the correct set of statements. Identify the correct set of statements. Just a minute बच्चे. So what should be the correct answer here? In 1997 the first transgenic cow Rosie was developed. So Rosie Didi produced human protein enriched milk. There are an estimated 20,000. Is it 20,000 varieties? The 27 documented varieties are grown in India only. Okay, so what should be the answer? Is it one? Is it one? Yes, of course it is one. है ना? Of course it is one. Chalo, now let's move to the next question. Human health and disease. Chalo, answer this question. Answer this question, and just a minute, people. After human health, we have very good number of questions of human health as well. Okay, so choose the odd one out. Choose the odd one out. Quick, choose the odd one out. Odd one out, people, odd one out. Autoimmune response, cell-mediated immune response, primary immune response, and then comes the physiological barrier. देखो, autoimmune में rheumatoid arthritis, CMIS, graft rejection means whenever there is organ transplant. Okay, the next is primary immune response. So primary immune response will not be anamnestic because that is secondary immune response. Okay, that is secondary immune response. So physiological barriers may killing of bacteria by the secretion, of course. Of course. Next question, which of the following does not hold true for the lymph nodes? Does not hold true for the lymph nodes? Yes, does not hold true for the lymph nodes. Is it a primary lymphoid organ? Arey, now speed is again fast. I need to go slow. I need to go slow. Chalo, I'll give you the break. Don't cry. Don't cry. I'll give you the break. So of course, it is the third. It is a primary lymphoid organ? No. When it comes to primary, you have bone marrow and thymus. You have bone marrow and thymus. Okay? Chalo, you need the break. I'll give you the break. Don't worry. So break time, break time, break time. Okay? Be happy. Don't cry. Break time, break time, break time. So break time is from. बच्चे देखो ना. Okay, so we started the class. So 12:20 to 12:40. That's sufficient, right? 12:20 to 12:40. That's fine. That's fine. Chalo, enjoy. Okay? Class is interesting, yeah? Class is not interesting. I am interesting. I am interesting. Skeezer बच्चे, now happy? चल smile कर दे फटाफट. Okay, चलो meet you after 20 minutes. Hello, hello, hello. All good? So can we start? Can we start? Okay. Now let's move to the next question. So guys, how's the energy? How's the energy? Now you got a break as well. So how's the energy? How's the energy? Quick. Quick. Okay, so how many diseases given below are not caused by bacteria? They are not caused by bacteria. Such type of questions will come in your paper. They are not caused by bacteria. Bacteria is not causing it. Yes, bacteria is not causing it. Quick, 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 quick. See, gonorrhea, bacterial, yes. Syphilis, yes. Genital warts, no. AIDS, no. Cholera, yes. Tetanus, yes. Typhoid, hanji, yes. Hepatitis B, no. Mumps, no. Herpes, no. Influenza, no. So 1, 2, 3, 4, 5. So, 6. So I missed something. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, and which one I missed? Tell me. Which one I missed? Quickly tell me. Tell me my mistake. Acha, not caused. Arey re, Ambika, वाह. देखा? Not caused, ठीक है. 1, 2, 3, 4, 5, 6. Wow, not caused. Dekha? So I am the live example that how to avoid the mistakes. How to avoid the mistakes. Even I am not reading the question properly. So I was like, bacterial diseases, mark it. So you can do the same mistake. Trust me on that. Trust me. है ना? Chalo, next question. Which of the following is incorrect about AIDS? Incorrect about AIDS. Quick. Incorrect about AIDS. Quick people, and share the link with your friends. Invite them, right? Invite them कि आ जाओ भाई वापस. Quickly come back. Yes. So which of the following is incorrect about AIDS? Is it a congenital disease? It's a congenital, माने by birth. Nope. It is caused by HIV. Yes. It is an enveloped retrovirus. Yes. You know, lipid envelope is there. The retrovirus enters into macrophages where RNA genome will replicate to form viral DNA with the help of enzyme. Yes. So it's 1. It's 1. Right? Next question. All of the following can be associated with the cancer except. All of the following can be associated with the cancer except. All of the following can be. Except. Activation of proto-oncogene into oncogene, suppression of tumor suppressor gene, activation of telomerase inhibitor, increase in production of telomerase. Now, you know na telomerase? What are telomerase? Tell me. What are telomerase? Tell me. Tell me. What are telomerase? Quickly tell me. What is telomerase? telomerase, you know na, chromosome, their end, right, their end, the telomerase enzyme will form that. You know that when a cell divide, the ends of that chromosome, they are that telomers now. So if they will keep increasing, it means the cell growth will be there. After many cell divisions, that ends they reduce, right? So in the cancer, the production of telomerase will increase. So the answer here is 3, right? It is not like that in the cancer, the telomerase inhibitors will not be there. The telomerase will produce, right? The inhibitors are not there, telomerase will produce. Now the next question, during initial days of lactation, abundant antibodies, they protect the infant and provide what are that antibodies going to provide? Yes, I know my students will answer the question. Please answer it, otherwise I'll get a heart attack. Yes, which during initial days of lactation, you are talking about the colostrum. So which immunity is it going to provide? Active or passive? Passive. Why? Because that baby is getting the ready-made immunity, ready-made antibodies, and it is the example of natural passive immunity. Tantana, right? Tantana, right? Next question. Read the following statement and choose the correct option. Read the following statement and choose the correct option. Yes. In allergy, the immune system does not lose the power of discrimination between self and non-self cells. B. Multiple sclerosis is an example of autoimmune disease. What should be the answer? Quick. What should be the answer? In allergy, the immune system does not lose the power. Obviously, allergy is just the exaggerated response of our immune system. It doesn't mean that they will stop recognizing self and non-self cell, that they will do in autoimmune disorder. So that is correct. So multiple sclerosis is an example of autoimmune disease. Yes, so the 1 will be the correct answer. Achha, you are saying 2 is the correct answer. Yeah, I am thinking 1 is the correct answer. Yeah, I am thinking 1 is the correct answer. What's going on? What's going on? There is some issue with our concepts then. What is going on? What is going on? So both the statements are correct. Next. Next. Select the incorrect statement about disease caused by protozoan. Answer it. Answer it quick. Yes, so incorrect statement. In malaria, hemozoin granules are responsible for fever. Yes. In amoebiasis, the magna form resides in small intestine. Arey nahi, that is magma, the pathogenic one, but it will not be there in small intestine. You will see it in large intestine. Is it caused by intramural histolytica? Yes. The malignant malaria is caused by falciparum? Yes. So second is our correct answer. Done. Second is our correct answer. Okay, now let's move to the next question. Match column 1 and column 2. Match column 1 and column 2. Be quick. Match column 1 with column 2. Quick. Quick, everyone. Physical barrier, physiological, they are making first line of defense, they are making second line of defense. So when it is the physical barrier, so we will be having the skin and mucus. So A with 4, फिजियोलॉजिकल में सलाइवा, सेलुलर में मोनोसाइट्स, साइटोकाइन में डर. So D with 1 we got our answer. Next, which among the following is an organ that is quite larger at the time of birth, but it will keep reducing in size with age, and by the time puberty is attained, it reduces to a very small size. It reduces to a very small size. Yes, it is the thymus. That's why with old age our immunity decreases. Our immunity decreases. Next, select the incorrect match with respect to the psychotropic drug. So I told you already, psychotropic are not the one that will cause hallucinations. The psychedelic drugs will cause hallucinations, right? The psychedelic drugs will cause hallucination. So what should be the correct answer? You need to figure out the incorrect match. Incorrect match. Quick. Incorrect match. Incorrect match. Yeah, some of you are mentioning 2, some of you are mentioning 4. I'm getting confused, right? I'm getting confused. Chalo. Even in the last class I told you that some chemicals, they even release the adrenaline and noradrenaline. So it will be done in, dekho, simple. Heroin, is it a depressant? Yes. Cannabinoids, principally receptors are there in brain? Yes. Cocaine, obtained from coca leaves? Yes. Nicotine, it stimulates the, dekho, adrenaline and noradrenaline will be released, but it will not be released from the cortex. It will be released from the adrenal medulla. So your adrenal medulla release catecholamines, right? They will release catecholamines. Is that clear? They will release catecholamines. Got it? Got it? Okay. So sometimes even a word can change our answer. So be careful when you answer the questions. Next question. Read the following statement about antibodies. So which of the above statements are correct? Which of the above statements are correct? Quick, which of the above statements are correct? I know you know the answer. I know you know the answer. Dekho, I already told you about the paratops. Your antigen is having epitop, your antibody is having paratop, and they both will join when there is the antigen antibody interaction, right? You know it already. Okay, you know it already. Secondly, if I talk about the same molecular weight, do you think it is possible? Because it is H2L2. 2 chains are heavy, 2 chains are light. Okay, so I'm giving you the hint. So according to you, what should be the answer? You have to figure out the correct statement, not the incorrect one. You have to figure out the correct statement, not the incorrect one. Not the incorrect one. So they are saying antigen binding site is present at N terminal. What is that N terminal? 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 So obviously the answer is D, right? The isotope is their isotype is determined by the constant region of the heavy chains. Okay, it's a fact when you talk about the other antibodies, right? So antigen binding site is present at N terminal. These are the correct statements. Clear, bache? Now the next question is select the set of disease which are vector borne. Which are vector borne. Select the set of disease which are vector borne. Quick. vector, vector, vector. देखो, pneumonia, droplet infection. I'm just picking up few points. Even common cold is also the same. AIDS maybe vector is not there, है ना? Or here also no vector is there. So second should be the answer. Malaria, chikungunya, dengue, mosquitoes, right? Mosquitoes. So destroy the mosquitoes. Next, all of the following diseases are transmitted through droplet infection except. Now, which one will not spread through droplet infection? Means like this, achoo. Okay, means some, even through the sneezing, yeah, in any form if saliva is coming in contact. So, it's simple, obviously bubonic plague, because here the vector is rat flea. Rat flea is your okay, spelling is Xenosylla chiopsis. Xenosylla chiopsis. Okay. So fourth, next, the use of vaccine and immunization programs have enabled us to completely eradicate a deadly disease called what is eradicated? Yes, what is eradicated? Totally. What is eradicated? Quick. The smallpox. है ना? The smallpox. Next. Very good. Allergy is an exaggerated response of the immune system to certain antigens present in the environment. It occurs due to देखो, don't you think that this option is not completed? है ना? Don't you think this option is not completed? देखो, heparin will not be released, the histamine is released, and it will be released from the mast cell. So I know the option is not, it is not mentioned completely, but still the answer will be this. So antihistamines, they are just taken when there is the allergic reaction. So that will not be the answer, right? Macrophages, they do not release the histamines. Histamines are released by your mast cells. They will tell the other non-infected cells ki bhai protect yourself, right? That is also the mast cells, they work. Next question, select the organ where B lymphocytes acquire antigen specific receptor, right? Where they will acquire antigen specific receptor, right? Select the correct organ where B lymphocytes require antigen. Yes bachche, B lymphocytes require antigen specific receptor. What should be the answer? Exactly, it is going to be 2, because what will be provided? Antigen specific receptor. And which lymphocytes you were talking about, right? The B lymphocytes, right? So it will be your, yes bachche, it will be your bone marrow. Now the next question. If a person is infected with some deadly microbe to which a quick immune response is required, the doctor must need to directly inject. Now quick response is required. We cannot wait for the body to act. So what will be added? Some microbe is there in our body. Quick response. The word is quick response. Yes. So obviously the preformed antibodies. So what type of immunity are they going to provide? Yes, it will be the passive immunity. है ना? And it will be the artificial passive immunity. Isn't it? Artificial passive immunity. Isn't it? Okay, next chapter, next question. Epitope epitopes are immunological active regions on where are these epitopes present? Where will you see the epitopes? Where will you see the epitopes? They are on antigen. So when you talk about antibody, antibody will be having paratop. Okay, antibody will be having paratop. Next question. Ringworm is caused by all except. Ringworm is caused by all except Monescus. Remember, Monescus, it is also a fungus, but it will give us statin. And yes, it is microsporum. Here it should not be macrosporum. So that is also a mistake if there will be the macrosporum here. Done. Next question. Which of the following drugs caused alteration in thoughts, feelings and perception without any actual cause? Wow. Wow. Alteration in thoughts, feelings and perception. Angie, what should be there? Alterations in thoughts, feelings and perception without any actual cause. Some are saying 2, some are saying 4, some are saying 1. Then mention the 3 as well. Huh? Without causing any actual cause. So obviously the answer will be marijuana. So they are asking about what? Somewhere the hallucinations. Somewhere the hallucinations. So marijuana will be the one. है ना? Wow. Next. All of the following options are associated with detection of cancer except. Yes. What should be there? It's simple. It's simple. Detection. The word is detection. Bache, radiotherapy will be given to treat it? No. Next, the drug which can be prescribed as a potent cough suppressant. I really need it. Cough suppressant. I need it. Cough suppressant. But a cough suppressant, it will suppress the cough. Guys, you don't know this? Cough suppressant, it will be codeine. It will be codeine. And do you remember in the biomolecules also in that table, this word is there. This word is there. It is not benzodiazepine. It is codeine. It is codeine. Is it incorrect? Is it incorrect? Is it incorrect? Tell me. If it is incorrect, deduct your marks. Come on, deduct your marks. Codeine, codeine. Okay. Okay. Next, which of the following is not useful in cancer treatment? Okay, not useful in cancer treatment. This one is very easy. Not useful in cancer treatment. -1. Learika, it's not -4, it's -1. Butterfly, butterfly, butterfly. So Kailash, what I need to do? Do I need to fly? Huh? Very simple. Antibiotics are not going to help. Antibiotics are not going to help. Which of the following can be considered as a method of prevention from transmission of AIDS? Hanji, method of prevention of transmission of AIDS. Quick. Quick. Method of prevention of transmission of AIDS. Quick. Obviously, the use of barrier method, condoms. Natural cannabinoid, ganja is obtained from ganja, है ना? It is obtained from your parents will kill me, है ना? Ganja, obviously hemp plant, it's a cannabinoid. From cannabis you are going to get it. So hemp plant. From this claviceps you will get LSD. You will get LSD. Next, all of the following methods can be used to diagnose HIV infection except very good, because this is for typhoid. Next, cancers of epithelial tissues are classified under the category of, this is also we have solved. Very good. Cancers of epithelial tissues are classified under the category of, everyone answer it. Seriously, Kailash, you really need to revise, bro. It's carcinoma. Next, in the given picture, A are the small solid structure located at different points along the lymphatic system. So they are quick. What are they? They are lymphatic nodes. Next, CNS stimulants which are obtained from coca leaves interfere the normal functioning of the limbic system by blocking. Remember, by blocking, we know the answer. Quick, what will they block? Good, very good, answer it. What will they block? Answer it. Exactly, that will be 1, reuptake of dopamine. Okay, reuptake of dopamine. Okay, so done. Next question, vinblastine. Why are, even this is what we have discussed in the last class. So vinblastine is the treatment, it is used in treatment of cancer, are included in. Remember, it is included in vinblastine. It is obtained from Vinca rosea. You all have seen Vinca rosea. It's a very common plant. Family is Apocynaceae. Exactly. It's a chemical. So chemotherapy. Next, which of the following pairs of innate immunity barriers belongs to the same category? Same category, simple. Exactly, this is the physiological barrier. Second, they are the physiological barriers. What are they? They are the physiological barriers. Now, read the following statement with respect to the aids and state them as true or false. Okay, read the following statement with respect to the aids and state them as true or false. AIDS is a congenital disease, means by birth. False. HIV attacks on T helper cells and macrophages because they have CD4 receptors. HIV, they will attack on the macrophages. CD4 receptors are there, but that we will see in the case of macrophages now. Okay? Chalo. So this B will be true. And macrophages cells can kill the HIV. Is it so? No, they will serve as the HIV factory. PCR can't use to detect the HIV in AIDS suspected patient. F. So what should be the answer? Is it 3? Yes, it is 3. It is 3. Next, antibodies present in colostrum contains. Wow, I like this question. I like this question. Antibodies in the colostrum contains. देखो antibodies in colostrum. Which antibodies in colostrum? You are giving the incorrect answer. Colostrum में you are having IgA. IgA is a dimer. I told you this is a good question. So if it is a dimer, how many heavy chains and the light chains will be there? How many heavy chains and the light chain will be there? The solution is 2. 2 heavy chain, the 4 heavy chains and 4 light chains. Bache, they are not stupid to ask such question na. It is actually a good question. I gave you the hint as well. Good question. Good question, because colostrum mane IgA. IgA is dimer, right? IgA is dimer, and when it is IgM, it is a pentamer. It's a pentamer. So I have seen that common sense is very rare these days. Common sense is not so common these days. है ना? है ना? मतलब ठीक है यार, they are just asking the number of chains. So 2 heavy, 2 light. Why यार? Right? Am I stupid who explains the topic like this? कि देखो बच्चों, J chain, secretory chain. This is what you have to remember. Okay? I'm not just reading NCERT in my marathon sessions, बच्चे. I'm explaining you the concept, and that is why I'm explaining you the concept. So please, 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 please be alert. Be alert. ठीक है? Otherwise, moe. Next. T and B lymphocytes are involved in cell-mediated and humoral immune system. Both these cells are produced from, yes, both of them are produced from, of course, it's easy. It's easy. Both the cells are produced from, quick. Some are saying 1, some are saying 2, some are saying 3. Then please even answer it as 4 also. That will be fun. Some will say 1, 2, 3, 4. Then I'll tell you which group is having the correct answer. Yeah, it's simple. Solution number 2. Solution number 2. Bone marrow. Both the cells, right? They are saying both the cells, they will form in bone marrow. Bone marrow mein basically the cells, the WBCs, they form in general. In general, they form in bone marrow. Right? Then further, the proliferation and all will be there in this bone marrow and the thymus part. Next, and even sorry, it is also a good question. It is also a good question. Okay, so next question, PMNL are blood cells which are a part of, yes, which are a part of, it is not a good question. It is not a good question. Which are a part of, it's easy, cellular barrier of immunity. The second line of defense and the non-specific type of immunity. Next, in life cycle of Plasmodium falciparum, the stage which infects the female Anopheles mosquito is the stage that infects female Anopheles mosquito. The stage that infects, dekha, again, it is also a good question. The stage that infects female Anopheles mosquito. Female Anopheles mosquito. Are yaar, gametocytes will be taken by the female Anopheles. No. Sporozoids are the infective stage of malarial parasite that will enter in our body. That will enter in our body. Chalo ji, agla question. Filling of alveoli with excess fluid, which reduces the diffusion rate of gases, causing bluish coloration to develop under nails, can occur due to I like this question. I will not say that it's a good question. So they just changed the way to ask it. Obviously, they are talking about Mr. Pneumonia. Who causes pneumonia? The Haemophilus influenzae, right? This bacteria. So that's why our answer is Mr. 2. So B lymphocytes on activation, they form dash, which produce antibodies against the antigen which has stimulated them. Okay. Okay. So B lymphocytes on activation form dash, that will form antibodies. Okay. Simple. Simple. Simple, simple, simple. It's simple. Obviously, the plasma cells, they form the antibodies, right? The plasma cells form the antibodies. Which of the following chemical is generally used to reduce the symptoms of allergy? Allergy. It will reduce the symptoms of allergy. It will reduce the symptoms of allergy. Which will reduce the allergy. Huh? I know you know the answer. I know you know the answer. What is the answer? What is the answer? Allergy. So obviously it will be adrenaline. And it will be adrenaline. Next, read the following statement and choose the correct answer. Benign tumors do not show metastasis. Morphine is very effective sedative and painkiller. So it is also a very simple question. It is a very simple question. It's a very simple question. Benign tumor do not show metastasis. Yes. Morphine is sedative and painkiller. Yes. So both are correct. Both are correct. Both are correct. Next, when a person is suffering from AIDS, the symptom due to opportunistic infection appears when? When? Very simple. It is also very simple. It is also very simple. Exactly, when number of T lymphocytes, the T helper cells, when it will be reduced, then other microorganisms, other pathogens will be like, let's have party in that particular human. They will also come there and they will start enjoying the weak immune system there. ठीक है? ठीक है बच्चे? Yes बच्चे. Next question. Okay. The chemical carcinogens present in tobacco smoke are the major cause of tobacco smoke. Say no to tobacco. Exactly, lung cancer. Bhai sahab, lung cancer. Tobacco smoke. Again, NCERT statement. Right? NCERT statement. Next, amphetamine. Amphetamine. Synthetic it is. Amphetamine. Amphetamine. Amphetamines. They are CNS stimulant drugs. They are CNS stimulants drugs. What are they? They are CNS stimulant drugs. Is that clear? Is that clear? They will stimulate the central nervous system. Next, nicotine is highly addictive as it stimulates the release of. Why this nicotine is addictive? Why nicotine is addictive? Quick. Why is it addictive? Because it will release Arey yaar, it will release adrenaline. And from where? From adrenal medulla. So adrenal medulla, they release the hormones which are amino acid derived, right? And what do we call them? We call them as catecholamines. Catecholamines. So next, choose the correct answer from the options given below. Health doesn't simply mean absence of disease or physical fitness. Balanced diet, hygiene, regular exercise are also good, important to maintain the health. So I think it is the simplest question that you can answer, right? It is the simplest question that you can answer. Fourth, what is it? Both are correct. Next. Next, coca alkaloid is obtained from erythroxylum coca plant. Coca interferes with the transport of dopamine. Again here, what should be the answer? Right? What should be the answer? Sanjani Singh, ma'am, how Madras transmitted? Bache, Madras is a disease. I am not aware of it. Obviously, both are correct. Next question. Choose the correct answer from the options given below. Virus-infected cells, they secrete proteins called interleukins. They are the protein protect non-infected plant from further viral infection. So what that viruses are going to release? Viruses, what will they release? Quick, what will they release? They are going to release interferons. So don't you think one will be the answer? Both are incorrect. Interferons will be released by virus infected cell. Next, mind and mental state can affect our health. Immune system maintains our health. Why yaar? Mind and mental state can affect our health, and immune system maintains our health. Obviously. Next, Plasmodium, a tiny protozoan, causes malaria. Chills and high fever occurring every 3 to 4 days in malaria. Yes, of course, both are correct. Next, prevention is best option for AIDS. AIDS has no cure. Again, it's a very simple one. Prevention is the best option for AIDS. AIDS has no cure. Both the statements are correct. Now, select the correct set of statements. Now, I like this question. Select the correct set of correct statements from the options given below. I know the answer. I know the answer. Quick. Even you know the answer. Trust me. You know the answer. There you go. Innate immunity is non-specific. True. Bacteria cells, they will release interferons. False. T lymphocytes mediate cell-mediated immune system. True. Vidal test. So 1, 2, 3. ACD. Next, correct set of statements again. Again, exactly. IgA antibody increase in allergy. True. As rheumatoid arthritis is not allergy, it is autoimmune. IgA antibody present in cholesterol. True. Injection of preformed antibodies. So again, fourth, nahi. Chalo, next. Again select the correct set of statements. Quick. Again select the correct set of statements. Quick, quick, quick. Again set the correct set of statements. Quick. Quick. Acquired immunity is pathogenic specific. Yes. Neutrophils and macrophages will form cellular defense. Yes. Humoral immunity is provided by B cells. Yes. Acha. Neutrophils and macrophages. Do macrophages will be the part of cellular barrier? Huh? Macrophages. Each antibody molecule has 4 polypeptide chains. Huh? So what should be the answer here? And tell me. Neutrophils and macrophages. Is it the macrophages, monocytes macrophages? Anji, so what should be there? Answer it. What should be there? Answer it. Fourth na? Matlab out of all options given here, right? So the most specific is ACD. ACD. Okay, let's go. So here ACD should be the answer. Okay. Next, morphin. Here. Answer it. Quick. Answer it. Morphin is a sedative. Yes. Cannabinoid is in brain. Opioid receptor are in CNS and GIT. Barbiturate are used as medicine. Done. So A, B, C and D. A, B, C and D. Bache, we are done with the break. Next, select the correct set of statements. Again, so same type of questions are there. Soon we will start assertion and reasons also. Dekho, what should be the correct answer here? Exactly, Haemophilus influenzae is responsible. Common cold is caused by rhinovirus. Hemocoin, no, it is him. What should be there? That word is not hemocoin. Virus infected cells will release interferons. So again, fourth is correct. Next, assertion and reason. Antibody mediated immunity is called the humoral immune system. The antibodies are found in the blood. So what should be the correct answer here? Quick. What should be the correct answer here? What should be the correct answer here? Dekho, antibody immunity, this is true. And the reason is the correct explanation of an assertion. Why do we say antibody mediated immune system is the humoral immune response? Because these antibodies, they are present in the blood, right? The humors, the blood, the fluid. So B should be second should be the correct answer. Next, opium is derived from the latex of unripe fruits of papaver. Excessive doses of drug may lead to coma and death. Okay, excess doses of this drug can lead to coma and death. What should be the answer? Dekho, both the statements are true, but do you think that reason is the explanation of the assertion? Of course not. So that's why 1 will be the correct answer, because reason is not the correct explanation of A. Understood? Next, in severe cases of pneumonia, the lips and the fingernails may turn grey to bluish. In pneumonia, the alveoli gets filled with fluid leading to problems in respiration. So what should be the correct answer here? Please read the options carefully. Please read the options carefully. Please read the options carefully. Exactly, right. Pneumonia mein lips and fingernails, they may turn grey in color due to the problems in respiration. So obviously reason is the correct explanation of A. Clear? Next, AIDS is not a congenital disease. During lifetime individual acquires it. Now what should be the correct answer? What should be the correct answer here? What should be the correct answer here? What should be the correct answer here? Of course, second. It is not a congenital, means by birth, because we acquire it during the lifetime. Simple. Next, which is not considered as a secondary sex organ. Oh my god, next chapter. Next chapter. Next chapter. People, are you ready? Ready for the next chapter? Human reproduction and then reproductive health. And the class will be over. So I think in next 1.5 hours, if you people will support, we can finish the class. Okay? So it's 1:35. We can finish it till 3:30. We can finish at 3:30. I'll give you a 15-minute break in between. Okay, so which one is not considered as a secondary sex organ? Testes. Why? Because they are the primary sex organ. They are the primary sex organ. Next, failure of testicular descent into the scrotum is known as. So here I will explain everything. Right, in the last class we were unable to revise the human reproduction now. So here I will explain everything to you. Dekho, it's very simple. When you talk about the testes, right, when you talk about the testes, bache, you know that, right, up to 7th month of pregnancy, they are abdominal, right? You know that the human gestation period is of 9 months. So after that 7th month, they will start descending into the scrotum. And how will they descend? They will descend through this inguinal canal, right? Through this inguinal, through this inguinal canal. So obviously the testes, they will descend into the scrotum, bache. And if testes are not descended into the scrotum, it will be cryptorchidism. It will be cryptorchidism. Okay, it will be cryptorchidism. Understood? Next, the structure through which testes descend, I just answered it. I just answered it. I just answered it. Quick. I want you people to answer this question. Be quick, guys. Be quick. Yes, what should be the correct answer here? What should be the correct answer here? I just answered it. It is inguinal canal. It is inguinal canal. Understood? Next chapter. Right, given below are two statements. In adult, each testis is oval in shape with a length of about 2 to 3 centimeter and width of about 4 to 5 centimeter, and they are having 250 compartments called testicular lobules. Quick. I'll revise everything with you. Definitely your confidence should increase. Guys, be quick. Saucy, Akash, Nandini, Dikshant, Sadhana, Kevin, answer this question. Answer this question, people. Answer this question. What should be the answer here? Seriously, bache, they are saying length is 2 to 3 centimeter, width is 4 to 5 centimeter. It is incorrect. Statement 1 is incorrect. Statement 2 is correct. Now let me explain you the statement 2. When you talk about the testes, they are present in scrotum and they are covered with the layers. You remember tunica is for the layer, and then you will say valve. What is that valve? Tunica vaginalis. Okay, tunica albuginea, tunica vaginalis, tunica albuginea, and tunica vasculosa. These are the 3 layers which are going to cover this testis. So now in these testes, right, just say I told you 3 layers are there. How many layers are there? How many layers are there? 3. So this tunica vasculosa, it will also cover these compartments which are present here in testis, and we call them that compartment as testicular lobules. What do we call them? We call them as testicular lobules. You remember my famous example, जैसे there is a building, let's say this building is having 250 floors and each floor is having 1 to 3 apartments. ठीक है, each floor is having 1 to 3 apartments. Is that clear? Is that clear? Right. So this is what you are going to get. 1 to 3 highly coiled seminiferous tubules. 1 to 3 highly coiled seminiferous tubules. Okay, so answer one of my questions, right? The seminiferous tubule is lined with. If you will answer this question to me, you people will get the chocolate, right? You people will get the chocolate. The seminiferous tubule is lined with. Yes, the seminiferous tubule is lined with. It is lined with. Very good, Samiksha bache. It is lined with the germinal epithelium. It is lined with the germinal epithelium. These are the cuboidal cells. It is the cuboidal epithelium. Very good. Very good. Now let's move to the next question. Right, let's move to the next question. The feature of some structures of male reproductive system is given below. Right, identify the structure on the basis of characters which surrounds the primary sex organ of male reproductive system. Which surrounds the male reproductive system. Male primary sex organ rather, which surrounds the primary sex organ of the male reproductive system. I know the answer. I know the answer. Quick. Quick. Obviously, testes, they are covered by scrotum. The thermoregulator will regulate the temperature, will lower it, will maintain the temperature 2 to 2.5 degree Celsius less than the normal body temperature, because that temperature is important for what? That temperature is important for your spermatogenesis. Your spermatogonia is temperature sensitive. For spermatogenesis, this is the temperature that we need. Next question. The male sex accessory ducts include. Accessory ducts. Obviously, it's not fallopian tube. Right, it's not fallopian tube. So your retetestis, your vasa efferentia, epididymis, right, vas deferens, they are making it. So of course, fourth is the correct answer. Fourth is the correct answer. Then study the statement given below and answer the question. Vigorous contraction of dash at the end of dash causes expulsion of fetus. I think anyone can answer it. Anyone can answer it. Obviously, vigorous contractions of uterus by the end of pregnancy causes expulsion of fetus. Expulsion of fetus is known as parturition. That is your childbirth. Parturition, that is the childbirth. So answer one thing to me. In the uterus, which muscles are present due to which there is a vigorous contraction? Answer this question to me. In the uterus, which muscles are present which are responsible for the vigorous contraction? Answer it to me. Which muscles are present in the uterus? Which muscles are present in the uterus? Muscles, I'm asking. Muscles. Arey yaar, smooth muscles. Smooth muscles that will undergo rhythmic contraction and relaxation. Smooth muscles. Excellent density. Excellent density. Myometrium is a layer, right? Myometrium is a layer, the middle layer made up of muscle. I asked the name of the muscle, right? Next question. Next question. Next question. The edge of the infundibulum possesses the finger-like, right? The finger-like projections called fimbriae, which helps in the collection of ovum. It's true. And let me tell you, it is the infundibulum which is closest to ovary, not the fimbriae. Fimbriae is just the extension of the infundibulum. Fun thing. Uterus is supported by tendons? No, it is supported by the ligaments. Okay, just ligaments, they attach. Ligaments, what are they going to attach? Muscle to muscle, and bone to muscle will be attached by tendons, if I'm not wrong. So statement 1 is correct, but the 2 is incorrect. So second will be the correct answer. Second will be the correct answer. Then next question, a very easy one. Yes people, it's a very easy question. I'm pretty sure you can answer the question. Match column 1 with column 2. Match column 1 with column 2. Hanji, I know the answer. देखो simple, menopause means cessation of the menstrual cycle. C with 2, ठीक है? And vagina, so now C with 2, 1 minute. Acha, you have many options where C is 2, okay? So endometrium is the site where implantation will occur. So your D with 1. Yes, how can I forget? Second is the answer, because endometrium is the site of the implantation. Then fallopian tube is site of fertilization. Vagina, where capacitation will be there, that is the charging of sperm. So second is the answer. Next, read the following statement carefully and choose the incorrect one. Read the following statement carefully and choose the incorrect one. It's a very simple question. Excellent. Shafna, Desiti, Priya, very good. Lakshmi, no, it is incorrect. Lakshmi, check the answer carefully. It's very simple, bache. Oxytocin, O for out. It is the milk ejection hormone. Right, bache, milk let down hormone. It is also the bonding hormone, the love hormone. Then secondary spermatocyte, you know that your spermatogonia, it will grow, it will form primary spermatocyte, that will undergo meiosis 1, you will get 2 secondary spermatocyte, that will further undergo meiosis 2, and you will have 4 haploid spermatids. So yeah, this is correct. Progesterone is anti-abortion hormone. It maintains pregnancy, so it is pregnancy hormone. So FSH surge is responsible for ovulation. No, it is luteinizing hormone. It is the luteinizing hormone, the LH surge. Next, read the following statement carefully and answer the questions given below. You need to find out the correct statements. Cyclic changes occur in endometrium during menstrual cycle. True. External thin membrane of uterus, okay, external, the perimetrium. Strong uterine contraction occur in myometrium at the time of parturition. True. Uterus opens into cervix through vagina. No, it opens into vagina through cervix. So this is false. So A, B and C. If you remember the structure, it will be like this. So here you will see the cervical canal, okay, the internal sphincter external, then there will be the vagina, okay, non-muscular, then there will be the vagina. Clear bache? Clear. So this statement is incorrect. Understood? This statement is incorrect. Okay, so yes, in uterus outermost layer is perimetrium, then myometrium, then endometrium. Myometrium will be having the muscles. Which muscles are there? Smooth muscles. Endometrium, it is glandular, right? It is basically secretory. It is the site where the implantation will occur, right? Next question. Read the following statement carefully and choose the correct options. Okay. Yes, You know the answer? No, Priya, you know the answer. People, please answer it. And guys, again I'm reminding you, do not forget here to enroll in your all India mock test. You will see the link in the description box of this particular video as well. Do you know one thing? I know some of you will say that in the coaching also you are having the mock test, right, from 2 to 5:20, then why should we enroll there, right? Sometimes maybe there the mock test will get cancelled there. You will be at your home, right? Then you can still practice the question. You will not waste your time there. Okay, you will not waste your time there. So definitely you should enroll yourself, and here it is for free, right? So, and yeah, the motto is not just to enroll, right? You have to give the test as well. Fine, you have to give the test as well. Now see, mammary gland is a modified sweat gland. True. PRL means prolactin. So who's going to release it? Anterior lobe of pituitary. It is responsible for production of milk from cells of alveoli. True. Nipples and its surrounding structure is macula. No, it is areola. Okay, so menstrual cycle do not occur during the period of intense lactation. This is called lactational amenorrhea too. So bache, in the final NEET examination, whenever you will attempt your paper, so make sure if they are asking correct and incorrect question, right, if they will ask correct and incorrect option, make sure you answer it like this. Now see, after marking statements like this, you can read the question again, then you can just highlight this word ki they are asking correct option. So 1, 2, 3. So A, B and D. That's how you can mark it. So you will not get confused. Okay, you will not get confused. Now correct match pairs are correct match pairs are correct match pairs are correct match pairs are. Yes, you know the answer, people. Quickly answer it. Clitoris, is it the erectile body in female? True. Colostrum is secretion of prepucial gland? No. Oxytocin, milk synthesis? No. It is for milk ejection. Labia majora is homologous to scrotal sac? True. So A and D will be the correct answer. A and D will be the correct answer. Clear? How come A and C? It is milk ejection, not milk synthesis. Synthesis will be done by prolactin, and even progesterone will be also play the role. देखो ना, prolactin, P production of milk, prolactin. So that's why 3 is the answer. Now next question. Okay, which of the following columns, and choose match them. I think we just discussed it, right? We just discussed it. So let's move to the next question. Yes, match this. Match this. देखो बच्चे, the correct answer, I know, you might not aware of these terms. Hysterectomy means removal of uterus. ठीक है? So A with A. Okay, when it is the orchiodectomy, it is the removal of testes. When it is the oophorectomy, देखो O word is there, ovary. Vasectomy, cutting off the vas deferens. Right? So sometimes elimination method can also help. Understood? So question can come from such terms. You should take a note of it. Okay, you should take a note of it. Understood? Next. Which of the following statement regarding parturition is correct? Regarding childbirth is correct. Regarding childbirth is correct. Sorry, incorrect, not correct, is incorrect. Very good, Desiti. Excellent, Desiti. Excellent, Shivani. Very good, Pradeep. Obviously, incorrect one. Prolactin induces uterine contraction. No, that will be oxytocin. That will be oxytocin. Right. Next question. Parturition is a neuroendocrine mechanism. The childbirth. Okay, the childbirth, Lakshmi, very good. Right, the childbirth. Now the signals for parturition originate from, they originate from, yes, they'll originate from. So guys, are you feeling energetic or are you feeling low? Ki nahi ma'am, we don't have energy, we just want to sleep. Dekho, you practice chemistry and physics question for 7 hours, for 8 hours. I'm not going to take it for that long because obviously biology, yaar, we have that benefit. Okay? So I think in next 40 minutes also we can finish it. Okay, so you before 3 let's finish it. What say? Let's not go for the break. Before 3 let's finish it. What say? Then there will be a break and break only. What say? Are you okay? Are you aligned? Let's finish it before 3. Then you can utilize that time for self-study, for revising it from the NCERT. And acha, do you want the quick revision session of organism and population? Tell me, do you want the quick revision session of organism and population? Jise for cell cycle and cell division, I don't know whether you have seen it or not, right? For cell cycle and cell division, ये देखो, see this. Jesse Wasim sir has also posted for electrochemistry, and I have posted it for the quick revision of your, see this, electrochemistry, and even for the cell cycle and cell division. So in 16 minutes we have revised the cell cycle and cell division. If you have checked it, very good. If not, please go for it. Okay, please go for it. And bache, same way, if you want me to make the sessions on organism and population, do let me know. Do let me know for the quick revision. Okay, so I'll try to post it today after the class. We'll try to post it today right after the class. Now, the signals for parturition originates from fully developed fetus only. Okay, fully developed fetus only. Clear, bache? Fully developed fetus only. Next. Placenta as well as fully developed fetus. Right? Placenta as well as fully developed fetus. Okay. Okay. Yes, my bad. Dekha again. Negative marking. Even I got the negative marking. Placenta and fully developed fetus. Next, identify the false statement about major features of the embryonic development. Quick. So see, through these MCQs we can revise the entire chapter. Quick. Identify false statement about the major features of the embryonic development. False statement. बच्चे second month, see देखो, they are saying by the end of second month of pregnancy the body is covered with fine ear eyelids. No, by the end of 12 weeks most of the major organ system will form. Okay, by the first trimester, first movement of fetus and appearance of hair on the head are usually observed during the 5th month. Correct. The first sign of growing fetus may be noticed by listening to correct. So they are asking the false statement. So 3 will be the correct answer. Okay, 3 will be the correct answer. Okay, so next, among the following, identify the structure which is involved in implantation. What will get implanted? Yes, what will get implanted? Which structure is involved in implantation? It's simple. Yes, the blastocyst. What that blastocyst will contain? This, right? So in the blastocyst there is a blastocele. First of all, this layer in the surrounding is the trophoblast. Okay, and here what are we going to see? We will see the cell mass. Okay, cell mass. So this is nothing but it is ICM, that is your inner cell mass. And it is the one that will form the future embryo. And the cells that come in contact with the, the cells of the trophoblast that are coming in contact with your inner cell mass, they are known as cells of Robber. right? They are known as cells of Robber. So basically this blastocyst is still there in the zona pellucida, right? The trophoblast cells, they will release the proteolytic enzymes, they will digest that endometrial lining, and what is going to happen? The implantation will occur. So 3 is the correct answer here. Next, okay, you know it. Identify the human development stage shown below as well as the related right places at its occurrence. Obviously, it is the blastocyst, we just discussed it. This is the blastocele, the cavity, and in the uterus it will be implanted. Okay, so if the implantation is at any place other than uterus, right, implantation should be in uterus, in the endometrial lining. If it is at any place, other than uterus, then that will be ectopic pregnancy. Is that clear? That will be ectopic pregnancy. Now, arrange the correct sequence of fertilization process. Yes, arrange the correct sequence of fertilization process. Quick, everyone, quick. Arrange the correct sequence of fertilization process. It will be fourth, right? The union of male and female gametes, intermixing of cytoplasm, fusion of pronuclei, and then finally the chromosomes will mix. Okay, chaliye next. Which one of the following enzymes facilitate the penetration of sperm through zona pellucida? It's simple. Penetration of sperm through zona pellucida. It's simple. Which one of the following enzymes facilitate the penetration of sperm through zona pellucida? It is acrosin, right? It is zona lysin, yeah, acrosin it is. Okay, next. A 30-year-old female goes to the doctor that after 4 days there is a due date of her menses and she wants to postpone the bleeding, right, for some days as she will be engaged in some official work. So which hormone the doctors are going to advise her to take? Simple. She wants to postpone her period. So which hormone doctors are going to advise her? Simple it is. Simple it is. Simple it is. A 30-year-old female goes to the doctor that after 3 days there will be the due date of her menses and she wants to postpone the bleeding for some days. Of course, she has to take the progesterone. You remember yesterday also I explained it, that progesterone in the body, it will give a signal to the hypothalamus that okay, progesterone is there, there is no need to release the gonadotropin releasing hormone. So that is how she will delay her periods. Okay, so it will be the progesterone. Okay, next question. If hysterectomy, you know that what is it? It is the removal, surgical removal of uterus. Okay, it is the surgical removal of uterus. If it is done in 25 years old female, which statement will be correct after the surgery? Yes, the uterus is removed basically. Hysterectomy means the removal of uterus. Answer it. Quick, everyone. Yes. देखो ना बच्चे, in the menstrual cycle what is going to happen? The answer here is 1. In the menstrual cycle what is going to happen? Basically endometrium lining will shed off, the egg, right? The basically देखो, in some hysterectomies sometimes even doctors they remove the ovaries, but if it is only the removal of uterus, that lady, that girl will not have the periods. The reason is now uterus is not there, endometrial lining is not there, that will not shed off. That will not shed off. Egg for, if ovaries are intact, hormones will be there, egg formation is there, but that proper periods will not be there. So 1 is the correct answer here. Next, LH act on Leydig cells, stimulate synthesis and secretion of androgens. FSH act on Sertoli cells, stimulate secretion of factors which help in the process of spermiogenesis. So which one should be the correct answer here? Which one should be the correct answer here? Yes. Which one should be the correct answer here? I am repeating this bache because we didn't revise it yesterday. So spermiogenesis, what is spermiogenesis? Right, what is spermiogenesis? Tell me. What is spermiogenesis? Spermiogenesis, also known as spermatoleiosis. Spermatoleiosis. When you are spermatid, it forms, it transforms into spermatids. When they transform into sperm, that is spermiogenesis. So what will happen here? What will happen here? Both the statements are correct. Both the statements are correct, because yes, LH will act on Leydig cells, right? The synthesis and secretion of androgens will be there, and FSH will act on the Sertoli cell, will stimulate the secretion of factors which will promote spermiogenesis. Next bache, right, given below are two statements. Statement 1 is given here, statement 2 is given. So statement 1 is saying that cyclic menstruation is an indicator of normal reproductive phase which extend between menarche and menopause. The first menstruation begins at puberty is called menarche. So what should be the correct answer here? Yes, what should be the correct answer here? Even it is a very simple question. It's a very simple question, guys. So we will continue the lecture till 3, and I hope you all are energetic. Let's answer it, bache. Let's finish everything. Okay, let's finish it. What should be the correct answer here? What should be the correct answer here? Obviously both the statements are correct, right? Menarche means the starting of menstruation, menopause when it will stop. So in between the menstruation is there and it will begin at puberty, and it is called the menarche. Clear bache? Right, so 4th is the correct answer. Now the next one, the blastomeres in the blastocyst are arranged in the inner layer called trophoblast. Outer group of cell is trophoblast and inner cell mass. Okay. The embryo with the 8 to 16 blastomeres is called blastula. So again you know the answer. Again you know the answer. Again you know the answer. Both the statements are incorrect. Why? Outer is trophoblast, inner is inner cell mass. And when you talk about the embryo with 8 to 16 blastomere, it will be It will be, what will be the word for that? Yes, what will be the word for that? That will be morula, isn't it? Morula. Next, HCG, human chorionic gonadotropin, human placental lactogen and relaxin are produced in women only during pregnancy. By the end of about 12 weeks, body of fetus is covered with hair, eyelids separate, eyelashes are formed. Now, what should be the correct answer here? Yes, what should be the correct answer here? What should be the correct answer here? Very good, Sharon. Sharon, excellent. Very good, Nandini. Krutika, Desiti, very good. Sharon, very good. So obviously, first statement is correct, but second is incorrect. So second is the correct answer. Second is the correct answer. Next, parturition is induced by a complex neuroendocrine mechanism. The signal for parturition originates from fully developed fetus and placenta. Right? This is also something that we have discussed right now, just now. Okay, so obviously here both the statements are correct, isn't it? Both the statements are correct here. Now let's move to the assertion and reason based questions. Let's move to the assertion and reason based question. So male accessory gland include paired seminal vesicle, a prostate and paired vulvourethral gland. The secretion of these glands constitute the seminal plasma, which is rich in fructose, calcium and certain enzymes. Yes, bache, what should be the answer, everyone? Liharika, the answer is incorrect. Samiksha, the answer is incorrect. Bache, in the male accessory gland include a paired seminal vesicle, a prostate and paired. It's true. Secretion of these glands will form are rich in fructose, calcium and certain enzyme. It is also true. But reason is not the correct explanation of A. Of course, it is not the correct explanation of A. Clear? Clear? So next. The presence or absence of hymen is not a reliable indicator of virginity or sexual experience, and hymen can also be broken by a sudden fall or jolt, insertion or a vaginal tampon. So what should be the answer here? What should be the answer here? What should be the answer here? So students, here the correct answer is 2, because assertion is correct, reason is correct, and reason is the explanation of assertion. Yes, but hymen can even, right, it can even be broken by a sudden fall or jolt. So insertion of vaginal tampon also, so we cannot say that it is the sign of virginity. Now let's move to the next question. Spermatogenesis starts at the age of puberty due to the increase in the secretion of vasopressin. After spermatogenesis, sperm heads become embedded in Sertoli cells and finally released from seminiferous tubule by the process called spermiation. It is very simple. It is very simple. It is very simple. Obviously, first statement is incorrect. It is not the vasopressin that will be released. Your gonadotropin releasing hormone, they will release FSH and LH, that will start it. So A is incorrect, but R is correct, right? So when from the Sertoli cells, when these sperm, they will be released in the lumen of the seminiferous tubule, yeah, we are going to call it as spermiation. We are going to call it as spermiation. Clear, bache? So menstrual flow results due to breakdown of endometrial lining of uterus and its blood vessel, which forms liquid that comes out through vagina. So ovulatory phase is followed by follicular phase. So what should be the correct answer here? Yes, Amrutha, what should be the correct answer here? What should be the correct answer here? What should be the correct answer here? Is ovulatory phase is followed by follicular phase? R is not correct. A is correct. A is correct. Clear bachche? A is correct, R is not correct. Next. During fertilization, a sperm comes in contact with zona pellucida layer of ovum and induces changes in the membrane that block the entry of additional sperms. So, mitotic division starts as the zygote moves through the isthmus of oviduct called cleavage. So, what should be the correct answer here? Read the statement carefully, okay? Read the statement carefully. Quick. What should be the correct answer? What should be the correct answer, students? Obviously, it is going to be 1. It is going to be 1. Both the points are correct, bache, but R is not the correct explanation of A. Okay, chalo. So again, I'll give you the break, right? I can see you people are tired. Or so from 2:15 p.m. the break time will be 2:30 p.m. Then in next 1 hour we will finish it. Okay, in next 1 hour we will finish it. Done? Done? So then after it you can do all that moi moi and all, and you can revise also. So we'll see you after 15 minutes. Hi everyone, welcome back. Welcome back everyone. So quickly share the session link and let's start, right? Let's start the session. Okay, so we have practiced many questions. Let's move to the next one. So please answer this particular question. You have to find the correct set of statements. Now I hope you all are feeling energetic. You all are charged enough to answer other questions as well. So let's finish it very quickly. Okay, we will finish it very quickly. So everyone just speed up. Speed up. Quickly share the session link with others and let's get started. Let's finish it ASAP. Let's finish it ASAP. So everyone, some more energy in the chat section, people. Some more energy in the chat section. Yes? Done? Yeah, I'm done with my lunch. What about you people? So what should be the answer here? The correct answer. The correct answer. Quick. They are saying the correct set. Each testicular lobule contain 1 to 3 highly coiled seminiferous tubules, right? The region outside the seminiferous tubule is the spaces. They contain small blood vessels and the interstitial cells. True. Male accessory ducts include the testes. No, no, no. Right? Accessory ducts, my testes will not be there. It is incorrect. Enlarged end of penis called glans penis is covered with the it is covered with what? Is covered with a tight fold. No, it will be the loose fold. Okay, so it is also incorrect. The secretion of bulbourethral gland, which is also known as Cowper's gland, help in the lubrication. So it's A, B and E. So what will be the correct answer here? Fourth will be the correct answer. Is that clear? Fourth will be the correct answer here. Now let's move to the next question, students. Identify again the correct set of statements. You need to identify the correct set of statements. Correct set of statements. Quick. Right, you need to find out the correct set of statements. Correct set of statements. Quick. It's very simple. Each ovary is about 2 to 4 centimeter in length. Each ovary is connected to pelvic wall and uterus by tendons. Of course not. Mons pubis is a cushion of fatty tissue covered by skin and pubic hair. Correct. The clitoris is a tiny finger-like structure which lies at the upper junction of 2 labia minora below the urethral opening. Is it so? Is it below the urethral opening? Nope. So glandular tissue of each breast is having 15 to 20 mammary lobes containing clusters of cells. So it's true. So the correct answer will be the third one here. It is the third one here. Now let's move to the next question. Again the correct set of statements. Right? Again the correct set of statements. Go for it. Go for it. Go for it. It's simple. You need to find out the correct set of statements. Quick, what should be the correct answer here? Kevin, answer it quickly. What should be the correct answer here? Each spermatogonium is diploid? Yes. The spermatids are transformed into sperm by the process called spermiation. No, it will be spermiogenesis. Okay, it will be spermiogenesis. Acrosome is filled with enzyme that help fertilization of the ovum. True. The secretion of epididymis, vas deferens, seminal vesicle and prostate are essential for maturation. True. Right bache, right bache. So the fourth will be the correct answer here. Right, it will be A, it will be C, it will be D. It will be A, it will be C, it will be D. Clear bache? Now what is the next question? Lactational amenorrhea, that is your lamb method, is based on the fact that ovulation and therefore the cycle do not occur during the period of intense lactation following parturition. Yes bache? So lactational amenorrhea method is based on the fact that ovulation and therefore the cycle do not occur during the period of intense lactation following parturition. IUDs decrease phagocytosis of the sperms within the uterus. So what should be the correct answer here? Everyone be quick. Right, let's finish it ASAP. So be quick. What should be the correct answer here? It's simple. It's simple. So they are saying that IUDs will decrease. Obviously this is incorrect. This is false. This is true. So statement 1 is correct. Statement 2 is incorrect. Next, Government of India legalized MTP in 1971 with some strict conditions to avoid its misuse. MTPs are considered relatively safe during the third trimester. Is it so? Is it so? Again, 1 is correct and second is incorrect. Right? 1 is correct, second is incorrect. Now, in amniocentesis, some of the allantoic fluid of the developing fetus is taken to analyze the fetal cells. So, it is used for the test for presence of certain genetic disorder, Down syndrome, hemophilia, sickle cell anemia. So, give me the most appropriate answer here. Most appropriate answer here, and trust me, you know the answer. Quick, everyone, you know the answer. You can answer it to me. Guys, be quick. Right, be quick. You can answer it to me. Obviously, statement 1 is incorrect. It should be amniotic fluid. Right, so A is incorrect. A is incorrect. R is correct. R is correct. So fourth is the correct answer. On what basis, Shaina, you are saying second is the correct answer? Right, it is not, it is allantoic fluid. Right, here it should be amniotic. Here it should be amniotic fluid. What should be the answer here? Amniotic fluid. Okay, next one. Read the following statement carefully. Yes, read the following statement carefully. Read the following statement carefully. So you need to figure out the incorrect statements. You need to figure out the incorrect statements. Quick. You need to figure out the incorrect statements. You can do it. In test tube baby program, embryo of 16 blastomere is transferred into the fallopian tube. No, this is incorrect. Art requires extremely high precision handling by specialized professionals and expensive instrumentation. True. PIDs, that is pelvic inflammatory diseases, stillbirth, infertility are some of the complications of STD. True. In vivo fertilization followed by embryo transfer into the female. No, it will be in vitro. So that is also incorrect. Right, intentional termination of pregnancy before full term is MTP. So the correct answer is A and D. Right, the correct answer is A and D. What will be the correct answer here? It will be A and D. Done? Okay, chaliye, now let's move to the next question. The head of the sperm contains the nucleus having a haploid set of chromosome in A. Right, the head of the sperm contains the nucleus having the haploid set of chromosome is A. Yes, having the haploid set of chromosome is the it is the, yes bache, bache, it is compact and it is in inactive state. So 1 will be the correct answer. 1 will be the correct answer. Next, placenta acts as an. Placenta acts as an. You know the answer, guys. You know the answer. Placenta acts as an. Yes, it is an, देखो, it is an endocrine gland, no doubt, but it is also an ultra filter. How? Because it will not let the teratogens that can cause birth defect to reach to the growing fetus, right, bache? So here it will be 3, both 1 and 2. Okay, both 1 and 2. Now, in humans, which of the following will undergo meiosis 2? Yes, which of the following will undergo meiosis 2? Which of the following will undergo meiosis 2? Guys, quick. Arey yaar, it will be secondary oocyte, but if the second polar body, if it is not degenerated, acha, हाँ, देखो, first polar body formed, firstly the secondary oocyte and the polar body. So secondary oocyte and the first polar body will go, right? First polar body will go. So here it will be 3. So if they will mention first polar body, then we have to select the first polar body and the secondary oocyte both, but they mention the second polar body. Okay, chalo, next question. Which of the following statement is not true? It is not true. It is incorrect. Which of the following statement is not true? It is incorrect. It is incorrect. Which of the following is not true? It is incorrect. Bache, incorrect. This one is incorrect. Not after the 1 month after fertilization, the blasto. Dekho, after 7 days of fertilization, it will get implanted. So this is correct. The divisions are rapid. Their divisions are asynchronous. Means the blastomeres, they will not divide at the same time. Clear, bache? Cleavage divisions are repeated mitotic division. So D is the incorrect one. Next. The base of the Sertoli cell adhere to the basal lamina and their apical ends frequently expand to the dash of the seminiferous tubule. Right? The base of the Sertoli cell is attached to the basal lamina, but their apical ends, right? But they are attached to the frequently expand into the, yes, into the lumen of the seminiferous tubule, right? Into the lumen of the seminiferous tubule. Understood? Chali, next question. So, intratesticular genital duct system includes the intratesticular, within the testis, right? Intratesticular genital duct system includes the intratesticular is the word. Intratesticular is the word. Right, so it is tubuli, recti, retitestis, ductili, efferentis. Okay, ductili, efferentis. Next, find out A and B respectively. Find out A and B respectively. What will be the A? What will be the B? They go simple. Anterior pituitary, LH and FSH will be released. Where are they going to act? Obviously on the gonads, and here because they are specifying estrogen and progesterone, it will be ovary, right? Then they will act on the uterus. So second should be the answer. Second should be the answer, right? Second should be the answer. Okay, next question. During the first 2 months of pregnancy, the basic structures are formed. During this period, the developing stage is called as, it's a very simple question. Quick. It's a very simple question. Quick. The developing stage is known as, bache, initially it will be embryo, it is not fetus. Initially it is embryo. It is not fetus initially, it is embryo. Next question. Which is not correct about the amnion? Which is not correct about the amnion? Not correct about the amnion. Not correct about, incorrect statement, right, about the amnion. Incorrect statement about the amnion. Bache, incorrect. It acts like a shock absorber. Yes. It provides a fluid medium to the developing embryo. Yes. It takes part in the formation of human placenta. No. So 3 should be the correct answer, right? It is the 3 which should be the correct answer. It is not going to take part in the placenta formation. Next question. Zona pellucida is synthesized by, again, it is the primary egg membrane. It's a very simple question. It's a very simple question. It is the primary egg membrane. It is the primary egg membrane. It will be released by, it's very simple. It will be released by, yes. Guys, stop chit chatting and please focus here, answer the question, and by the end of the class, do let me know your score as well. You should mention your marks today in the comment section. Okay, right? And you know that what you have to enroll, I will keep reminding you, AIMT, do not forget about it. Okay, do not forget about it. So zona pellucida, right, it is synthesized by oocyte, and if you talk about the corona radiata, that will be by the follicular cells. Next question. Corpus luteum is formed by, yes, corpus luteum is formed by exactly the granulosa cells and the thecal cells. The granulosa cells and the thecal cell. Granulosa and follicular cells are same, so the answer will be granulosa cells and the thecal cells. Which of the following set is correct? Which of the following set is correct? Quick. Which of the following set is correct? Quick. Which of the following set is correct? Exactly, all 4 are correct. All 4 are correct. Next, which pair is correct? Which pair is correct? Which pair is correct? Again a simple question. Again a simple question. Bache, whenever you talk about the sex hormones, they are steroidal in nature. Right? What are they? They are steroidal in nature. And if you talk about a cell, their receptors will be within the nucleus. Okay, so they will be having intracellular receptors. What do they have? They will be having intracellular receptors. Okay, so here the third will be the correct option. Next. Which of the following method of contraception is effective only up to a maximum of 6 period following parturition? Contraceptive method. Contraceptive method. Right? Right? So it is lactational amenorrhea, because during this time prolactin production is there, FSH and LH will not be there, right bache? Next question, which is included in the barrier method? Which one is included in the barrier method? You need to increase your speed, right? You need to increase your speed. The questions are very simple, and the answer, after reading the question, answer should be there in your mind. Immediately you need to answer it. Of course, all of above, right? Next, which of the following statement is correct? Quick. Which of the following statement is correct? Which of the following statement is correct? Hormone releasing IUD make uterus unsuitable for implantation. IUDs are ideal contraceptive for female who want to delay pregnancy. IUD is most widely accepted method of contraception in India. So all of above. So fourth will be the correct answer. Fourth will be the correct answer. Next, which is correct for Saheli? Saheli. Is it a weekly pill? Yes, bache. Is it having high contraceptive value and least side effect? Yes. Non-steroidal preparation is there, centromane or meloxin or meloxifene. Okay, so all of above. Again, all of above. Understood? Chalo, move to the next question. Which of the following can be used as an emergency contraceptives to avoid the possible pregnancy? Emergency contraceptives to avoid the possible pregnancy. The word is emergency contraceptive, so that it can avoid the pregnancy. Bache, emergency may obviously the diaphragm, IUDs, they will not be preferred. So, right, can be used in an emergency? No, 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 no. No, they are not for emergency, right? Nope, not at all. Not at all. They are not for emergency. they are not. हाँ ठीक है, देखो progestogens can be used, progesterone hormone, it can be basically what we have to do, emergency contraceptive to avoid possible pregnancy. No, so I need to check the options here, right? I need to check the options here. It is not possible. IUD can still be used within 72 hours, but not like this, right? Here for emergency, you remember I pill and all, they'll be preferred, right? They'll be preferred. IUD can still be preferred, right bache? So emergency contraceptives mein, yeah, hormones will be there, so progesterone and IUD. Okay, one will be the answer. Diaphragm will not be there. IUD within 72 hours, okay, because they will also start their function, they will not let the pregnancy to occur, so theek hai, fourth will be there. Now, which is highly effective but having poor reversibility? This question is giving us the hint. Highly effective but having poor reversibility. Highly effective but poor reversibility. Obviously sterilization, the tubectomy and vasectomy. Okay, the tubectomy and the vasectomy. Understood? Next question, which of the following statement is correct? 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 Guys, quick. Exactly, all four are correct. They are having significant role in decreasing the one second. MTB is safe during first trimester. Government of India legalized in 97. It is having a role in decreasing population, but it is not used as a method for decreasing the population, right? It is not used as a method for decreasing the population. So couple unable to produce the children in spite of unprotected sexual cohabitation is referred to as, they are termed as, right? They are not able to give birth to the babies even after having unprotected sexual intercourse. It is infertility. Next, infertility cases due to inability of male partner to inseminate the female corrected by. Dekho, here they are specifying that male partner is not able to inseminate. Then what can be preferred? Very good, Leharika. Very good, ACH, Meera. Yes, Monisha, Samiksha. Very good. So it will be artificial insemination. What will be there? Artificial insemination will be there. Okay. So in which of the following methods zygotes or early embryo up to 8 blastomere? Again, the direct question. Up to 8 blastomere is the word. Up to 8 blastomere is the word. Quick. Up to 8 blastomere is the word. Of course, it will be ZIF, that is zygote intrafallopian transfer. Is that clear? That is zygote intrafallopian transfer. Okay. Next, arrange the components of mammary gland. Right? Arrange the components of mammary gland. Arrange the components of mammary glands. Arrange the component of mammary glands. What should be the answer here? Yes, what should be the answer here? So here it will be alveoli. They will together form mammary. And the alveoli, what are they going to form? वैसे तो we know that alveoli all together they will form the lobes, but lobes are not mentioned here. So it will be the tubules, then there will be a duct, then duct will form ampulla, and finally lactiferous duct. So B, B should be there at the end. So 3 is the correct answer. It's the 3 which is the correct answer. Next question, assertion and reason based question. During pregnancy, the level of thyroxine is increased in the maternal blood. Pregnancy is characterized by metabolic changes in the mother. Yes, then what should be the answer here? Yes, people, tell me what should be the answer here? What should be the answer here? Exactly. देखो, no doubt during pregnancy the level of thyroxine is increased in the maternal blood, because here the metabolic changes will be there in the mother. So 1 will be the correct answer. Then how many secondary spermatocytes are required for forming 200 million spermatozoa? It's simple. It's simple. What should be the answer? What should be the answer? How many secondary spermatocytes are required? How many secondary spermatocytes are required? Exactly. देखो, it's simple. 1 primary spermatocyte will give you 2 secondary spermatocyte, and 2 secondary spermatocyte will give you 4 spermatids. ठीक है? So 1 will give you 2. So you need 400 million, right? So you will be needing 200, right? 1 will give you 2, so 200 will give you 4. So 200 should be there. 200 million should be there. So 3 should be the correct answer, but here it is marked incorrectly. It should be 3. Let me write it. It should be 3. Okay? 3. Then select the correct sequence. Select the correct sequence of transport of sperm cells in a male reproductive system. Yes? What should be the answer here? Exactly, it's seminiferous tubule, then turreted testis, vasa efferentia, epididymis, vas deferens, ejaculatory duct. No. Definitely not. Next, extrusion of second polar body from the egg nucleus occurs after. It's a repeated question. Extrusion of the second polar body from the egg nucleus occurs after. Yes, it occurs after. It occurs after. Obviously after entry of sperm, but before fertilization. Okay, but before fertilization. Next, no new follicles will develop in the luteal phase of the menstrual cycle, because no new follicle will develop in the luteal phase of the menstrual cycle, because in the luteal phase, what's the scenario? What will be there? What will be there? Yes? Obviously, because both of them, they are low here in luteal phase. Luteal phase means secretory phase. So in that phase, what will be happening? Your progesterone level is high. So 4th will be the correct answer. 4th will be the correct answer here. Clear, bache? Next, which of the following hormone level will cause release of ovum from Graafian follicle? So I think it is the easiest question. It is the easiest question, right? Which of the following hormone levels will cause release of ovum from the Graafian follicle? Hanji. They go out of all the options given here, you need to select it. Right, out of all the options given there, you need to select it. Of course, so here high concentration of estrogen. So even your LH surge will cause the ovulation, and that time estrogen is also high. Next, match the following questions. Match the following. Hanji, match the following. Quick people, match the following. They go C with the 4 directly. So cancel these two options. Then A placenta, HCG या फिर A placenta layer of the ovum. Obviously A with the 2. A with the 2. Now next, meiotic division of the secondary oocyte is completed. Meiotic division of the secondary oocyte is completed. Yes. At the time of fusion of sperm with an ovum, of course, ma'am. So receptors for sperm binding, they are present in. Receptor for sperm binding, they are present in. This is important MCQ, right? It is an important MCQ. The receptor for sperm binding, they are present in. They are present in zona pellucida, which is an acellular layer, and this is known as the primary binding. What's that? It is known as the primary binding. Is that clear? It is known as the primary binding. Clear बच्चे? चलिए next question. Which of these is not an important component of initiation of parturition? Initiation of parturition. Childbirth. For childbirth, which hormone is not required? For childbirth, which hormone is not required? Which hormone is not required? Definitely prolactin, because it helps in milk production, right? Because it helps in milk production. Is that clear? Because it helps in milk production. Next. So which of the following secretes the hormone relaxin during the later phase of pregnancy? Which of the following release the hormone relaxin during the later phase of pregnancy? During the later phase of pregnancy. Who's going to release that? Who's going to release that? Of course, the answer here. Corpus luteum also in the later stage of pregnancy, it will release the hormone relaxin, right? Then, statement based question again. The statement based question again. So what should be the answer here? Yes people, what should be the answer here? What should be the answer here? The release of sperm into the seminiferous tubule is spermiation. True. Spermiogenesis is the process of formation of sperm from spermatogonia. No. Spermatid will form sperm. Okay, that is spermiogenesis, also known as spermatoleiosis. So one is correct, second is incorrect. Next, the contraceptive Saheli. What will that do? It's also a PYQ, guys, so I'm pretty sure you know the answer. The contraceptive Saheli, the weekly pill, the having non-steroidal preparation, what is it going to do? Yes? Exactly, it will be one. It will not allow the implantation to occur. The estrogen receptors will be blocked. Okay, it will be blocked. Clear bache? Next question, select the hormone releasing IUD. 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 Exactly, progestasert and LNG20. Next, which of the following contraceptive method do involve the role of a hormone? Yes, do involve the role of a hormone. एग्जैक्टली देखो, बैरियर मैथड नो हॉर्मोन्स, CUT नो हार्मोन, ठीक है, बैरियर मैथड नो हॉर्मोन्स. So lactational amenorrhea में hormones are there, it's the natural method. Then pills and emergency contraceptive में also hormones are there. Okay, so solution is 1. So which of the following is a correct statement? Which of the following is a correct statement? Now answer this question, people. Which of the following is a correct statement? Which of the following is a correct statement? So they will increase the phagocytosis. Obviously, they'll increase the phagocytosis. Then, in which of the following techniques the embryos are transferred to assist the females who cannot conceive? Right, embryos are transferred. Yes, people, embryos are transferred to assist those females who cannot conceive. Right, who cannot conceive. Right, embryos are transferred, that's the word. So if it is the transfer of embryos, so it will be fourth, ICSI and just a minute, embryos are transferred, sorry, not the ICSI, it will be second, ZIFT and IUT. Here up to 8 blastomere, and if it is above then it will be intrauterine transfer. Okay, it will be intrauterine transfer. Next, which of the following is an example of hormone releasing IUD? Which of the following is an example of hormone releasing IUD? Hormone releasing IUD. Yes, hormone releasing IUD. I am pretty sure you can answer it. It is LNG20, and these three are copper releasing. Next, venereal diseases can spread through. Venereal disease means reproductive tract infections or sexually transmitted infections. So they will spread through, spread through. It is also a PYQ, guys. It is also a PYQ. Okay, so solution is 3 here, BC, not with the kissing. So it was also a confusion, right? While paper solving, I still remember it was also the confusing option. It was also the confusing option, right? Next, match list 1 with list 2. Yes, people, match list 1 with list 2. Match list 1 with list 2. Quick, list 1 with list 2. So vasectomy, tubectomy, C2D4. So A with 1, B with 3. Simple, second is the answer. Next, lips loop is a type of contraceptive used as, lips loop is a type of contraceptive used as, yes bache, it is used as non-medicated IUD. Then again, match list 1 with list 2 with respect to method of contraception and their respective actions. Again, it's very simple. Yes, it's very simple. So, देखो, lactational amenorrhea, absence of menstrual cycle, IUDs, increased phagocytosis of sperm within uterus. Right, diaphragm, they cover the cervix, blocking the entry of the sperm. So contraceptive, B with 1. So you got this as your answer. This is the 1. 1 will be your answer, right? 1 will be your answer. Okay? So which of the following is not an IUD? Not an IUD. Progestogen, isn't it? Progestogen, it is not an IUD. So tell me, up to this, up to, I think we have solved more than 350 questions. So tell me, what's your score? So out of, if I consider it as 7, if I consider it as 450, so how many, sorry, 350, how many questions you marked correctly? How many questions you marked correctly? Tell me quickly. And guys, I'm waiting for your comments as well, right after the class. I'm waiting for your comments, and make sure you check these videos. And yeah, it is today the last day of your Unlock 20 as well. If you want to enroll in any of the NEET UG courses, you guys can. Okay, so up to this, tell me how many questions are correct, how many questions are correct, and then answer the next one, 13 are incorrect. 13 are incorrect. Then Shivam, start revising them. Chalo. Next question. IUDs are small objects made of plastic or copper that are entrusted in the uterine cavity. Which of the following statements are correct about IUDs? They don't decrease. Okay? So cancel this option, elimination method. The released copper ions suppress the sperm motility. They do not make the cervix hostile. They do. So C, cancel. IUDs suppress the fertilization capacity. Okay. IUDs require surgical intervention for their insertion. Not exactly the surgical one. So what should be the answer? Can we say that it is third, B and D? Of course we can say so. Now, match the following disease was the causative organism. With the causative organism. Quick. With the causative organism. Rest are bache again from human health and disease that we have already practiced. Okay. Acha. So typhoid, Salmonella typhi, pneumonia, Haemophilus, filariasis, Vegeria, malaria, Plasmodium. Okay, so that's it, that's all. Right, so see you all soon in the next marathon. Right, in the next marathon you guys will be updated. So tomorrow your HSP sir is going to take the class, so make sure you like his video. Right, so you make sure you like his video. Okay bache, so he'll be taking your complete class 11th organic chemistry. So I'll be waiting for the comments. Right, and please check the yesterday session as well for your quick revision. So see you all soon, and in these marathons we'll complete the syllabus and we'll ensure your 340 plus marks, because we are also practicing the questions now. So you tell me, how was the lecture? It was good, right? It was good. Chalo. We will complete the evolution in the botany part. Why are you getting worried? So take care guys, we'll share the PDF. So bye bye, take care, thank you so much everyone.